The first sign that something is wrong is not dramatic. It is a label on a plastic bag, a sequence of letters typed into a hospital computer, a small line in a laboratory report that most people never need to read closely. Outside the emergency department, rain darkens the road and turns the ambulance lights into smeared flashes of red across the glass. Inside, a patient is losing blood faster than the body can replace it. The room is controlled urgency gloved hands, clean metal, cut fabric. The low precision of people trained to make decisions before fear has time to speak. Blood pressure drops. The patient's skin cools. Someone calls for units of red blood cells, because blood is no longer an idea here, no longer a symbol of family, ancestry, or secret inheritance. It is oxygen delivery. It is time. It is the fragile red current separating a living brain from silence. In moments like this, hospitals may reach first for one of the most valuable resources in medicine, negative red blood cells. They can be given rapidly in many emergencies before a patient's full blood type is confirmed, because they lack the A and B markers that can provoke the most immediate and violent incompatibility reactions, and they also lack the major RH marker known as D. The bag enters the room with no aura around it. There is no ancient crest stamped on its surface. No visible proof of mystery. It is simply blood collected from an ordinary donor who walked into a clinic, rolled up a sleeve, and left behind something that may now prevent another human being from dying. Yet the little negative sign written beside the letter O carries a peculiar power. It tells the doctors that this blood is useful in an emergency. It tells the blood bank that supplies must be guarded carefully, and beyond the hospital, it has become the seed of a far stranger story. A negative blood is often spoken about as though it is a biological riddle hidden in plain sight, a mark of forgotten ancestry, a survival of some lost people, even evidence of a human line set apart from everyone else. In the darker versions of the tale, it is treated as something science refuses to discuss openly, an inconvenient clue concealed behind routine medical language. The truth begins in a far less theatrical place, but it is no less powerful for being real. Odd negative blood matters because the immune system notices difference. A person whose red blood cells do not carry the D antigen may live an entire life without knowing it, never feeling different, never sensing anything unusual in the pulse at the wrist. But if that person is exposed to red cells that do carry DS through transfusion or pregnancy, the immune system may begin to learn the shape of what it sees as foreign, and once it learns, it can remember. Before we go deeper, take a moment to like the video and subscribe, if this is the kind of history and mystery you enjoy. Because the trail begins not in a ruined temple or a sealed tomb, but in the almost invisible surface of a red blood cell, where a feature too small to see has shaped medicine, childbirth, survival, and an extraordinary collection of myths about who we are. The patient in the emergency room does not know any of this. Perhaps the patient is unconscious. Perhaps the family is being kept behind a door while the surgical team searches for the source of the bleeding. In the blood bank, technicians move with a different kind of urgency. They confirm labels, check compatibility, track available units, and watch the dwindling stock of the blood that can be used when there is no time to wait. O-negative blood is not common, and it cannot simply be spent without thought. Every unit released now may be the unit needed an hour later for a woman hemorrhaging during childbirth, for a child brought in after an accident, or for a patient known to be Rh-negative, whose body must not be exposed unnecessarily to Rh-positive red cells. Medicine has learned, sometimes painfully, that compatibility is not an administrative detail. It is part of survival itself. To understand why, it helps to strip away the mythology for a moment and imagine blood not as a single red fluid, but as a moving population of cells, each carrying microscopic structures on its surface. And these structures are called antigens. They are not messages about destiny, intelligence, or hidden capacity. They are molecules that the immune system can recognize. The familiar blood groups A, B, AB, and O describe one important collection of these markers. The positive or negative sign, usually added afterward, describes whether the major RhD antigen is present. A person labelled A positive has A-type markers and the D antigen. A person labelled A negative has the A markers but lacks D. The same principle applies across the familiar blood types. That minus sign is not an exotic substance added to the blood. It usually indicates the absence of one particular marker that most people possess. Absence, however, can matter enormously when the immune system is involved. If Rh-negative red cells circulate inside an Rh-negative person, nothing unusual occurs. They belong there. But if D-positive cells enter that person's bloodstream, the immune system may identify the D antigen as foreign and begin producing antibodies against it. This does not always create immediate disaster at the moment of first exposure. That is one reason the danger remained hidden for so long in human history. The first encounter can pass without a visible crisis while the immune system quietly prepares. Later, if D-positive cells appear again, antibodies already waiting in the blood may bind to them and mark them for destruction. Red cells meant to rescue the patient can instead become targets. The body, trying to protect itself, begins attacking the very oxygen-carrying cells it has just been given. There is a cruel subtlety in that process. Blood looks the same in the bag. Under ordinary light, Rh-negative and Rh-positive blood offer no visible clue of the distinction. For most of human history, physicians could see bleeding, fever, weakness, jaundice, shock, and death, but they could not see the molecular incompatibilities beneath them. Early transfusion was haunted by unpredictability. Some patients survived, others deteriorated catastrophically. Even after the ABO groups were understood in the early 20th century, complications still appeared that did not fit neatly into the new system. The evidence remained incomplete until researchers recognized that human blood carried more identities than A, B, and O. One of the most medically important of those hidden distinctions became known as RH. The name itself has encouraged more confusion than it deserves. It grew from early experimental work involving rhesus monkeys, and over time the label RH remained attached to a complex human blood group system. But the name does not mean Rh-negative people are somehow less connected to ordinary human biology, nor does it reveal an alien or supernatural inheritance. It is a historical name for a real set of antigens and genes. The most famous of them is D because it is especially important in transfusion medicine and pregnancy. When people casually say someone is Rh positive or Rh negative, they are generally referring to this one antigen, although the broader Rh system is more complex, with several related antigens that can also matter in medical care. In the emergency room, that complexity is compressed into decisions made at speed. The bleeding patient needs red cells now. A properly selected unit buys minutes, stabilizes circulation, keeps the heart supplied, keeps the brain alive, but blood banks must also think beyond the immediate crisis. Giving Rh positive blood to an Rh negative patient may create antibodies that follow that person for years, in a man or in a woman beyond childbearing potential. A hospital facing extreme scarcity may sometimes weigh urgent needs differently, depending on circumstances and local practice. In a young Rh negative woman, the stakes carry an additional shadow. If she later becomes pregnant with an Rh positive fetus, antibodies formed after earlier exposure can cross the placenta and attack the fetus's red cells. A decision made in an emergency can echo far beyond the hospital room. For generations before modern prevention, that echo could be devastating. A healthy woman might carry her first pregnancy successfully, never knowing that fetal cells entering her circulation had sensitized her immune system. During a later pregnancy, if another fetus carried the D antigen, her antibodies might cross the placenta and begin destroying the baby's red cells. The result could be severe anemia, heart failure, jaundice, brain injury, stillbirth, or a newborn desperately ill from a conflict no one could see with the naked eye. Families once experienced these losses as baffling repetition, a first child thriving, later infants sickening or dying, grief returning without explanation. There was nothing mystical about it and nothing deserved. It was immunology operating without warning in a time before medicine knew how to interrupt it. That history is one reason Rh-negative blood accumulated such emotional weight. A trait carried quietly in the blood could influence who could safely receive a transfusion and which pregnancies faced danger. It could appear in family patterns, pass from parent to child, and remain invisible until a moment of crisis. Anything inherited, rare, and consequential naturally invites storytelling. Human beings are not inclined to leave such facts alone. A difference that affects birth and death quickly becomes more than a laboratory observation. It becomes a sign, a question, perhaps a warning, and where the scientific explanation is unfamiliar, fragments of fact begin to mingle with suspicion. The mystery deepens because Rh-negative blood is genuinely uncommon in many populations, though not equally uncommon everywhere. In some countries and ancestry groups it is distinctly rare. In parts of Europe it is encountered more often. That uneven pattern has led many people to assume there must be one dramatic origin story waiting to be uncovered: a lost tribe, an ancient elite, an isolated human lineage. Something that can explain why the negative sign appears more frequently in some places than others. Population genetics can indeed illuminate inheritance, migration, isolation, and chance. But rarity alone is not evidence of hidden power, and geographic concentration alone is not proof of a secret bloodline. Human populations carry many traits unevenly distributed across the world. Some are visible in hair, skin, or facial form, others remain silent until a test reveals them. Rh status belongs to that quieter category. Still, it is easy to understand why the imagination reaches for grander explanations. The ordinary account sounds almost too small for the consequences involved: a molecule present or absent on a cell, an immune response forming after exposure, a compatible blood bag arriving at the right moment. Yet within that small mechanism lies an entire human drama. It includes donors whose names the recipients never learn, infants once lost before the cause was understood, mothers screened routinely because knowledge now prevents tragedy, and hospital shelves where certain blood types are guarded like a reserve of borrowed time. It includes the deep unease created whenever something inside the body seems capable of dividing people into categories with life or death importance. In the operating room, the patient's numbers finally begin to improve. A transfusion is not a miracle in the old sense. It is the result of accumulated human attention, the discovery of blood groups, the discipline of matching, the development of testing, and the decision of a stranger to donate. The bleeding slows, oxygen begins reaching tissues more reliably. A line on a computer screen turns from alarming to cautiously hopeful. No one in the room speaks of ancient lineages. No one needs to. The clinical reality is already powerful enough. A missing antigen, invisible and silent through ordinary life, has helped determine which blood could be given safely when seconds mattered. And yet, beyond these walls, the negative sign keeps attracting other meanings. It appears on websites promising forbidden history, in stories of unusual abilities, in claims that those who carry it descend from beings or peoples unlike the rest of humanity. Some of these stories begin from real medical facts, then stretch them until medicine itself disappears beneath the legend. The tension at the heart of the enigma is born here. Rh-negative blood is neither meaningless nor magical. It is a genuine biological variation with serious consequences in particular circumstances. It is rare enough to provoke curiosity, significant enough to have shaped medical history, and silent enough in everyday life to invite mysteries into the gaps. The question, then, is not whether Rh-negative blood matters. It clearly does. The question is how an invisible feature of ordinary human blood came to bear so much medical importance, why it appears with striking differences among populations, and how those facts became entangled with stories of ancient secrets and concealed truths. Long before the theories, long before the whispered claims of special descent, there is only the bloodstream itself, red cells passing through darkness. Carrying oxygen, carrying inherited variation, and carrying a small absence that the immune system can turn into memory. That small absence does not begin in a hospital, and it does not begin with anyone knowing it exists. Its story reaches backward into an age before records, before cities, before anyone could imagine that the warm red fluid inside an injured body carried microscopic signatures from distant ancestors, somewhere in the immense and largely unrecorded movement of early human life, among populations surviving cold seasons, difficult births, infections, famine, predation, accidents. A migration across unfamiliar ground. Genetic variations appear and are passed onward. Most vanish almost immediately, lost when a carrier leaves no children, or when a tiny family line ends without ceremony. Some remain rare. Some spread because they provide an advantage, others spread for no grand reason at all, carried by luck through small groups whose descendants multiply while neighbouring groups disappear. Rh-negative blood belongs to this deeper world of inheritance, where biology does not always preserve a clear story, and where survival is shaped not only by adaptation, but by accident, isolation, and time. To imagine the beginning of such a trait, it is necessary to step away from the modern habit of looking for purpose. A mutation does not appear because a population needs it. It is not summoned by a harsh winter, a disease outbreak, or a dangerous migration. Genetic changes arise as part of the ordinary copying and reshuffling of DNA from generation to generation. Most have little visible effect, some are harmful, a few become useful in particular environments. But a variant can also remain in a population simply because its carriers survive and have children, and because the group around them is small enough for chance to matter enormously. In the deep human past, chance often had a louder voice than it does in modern populations of millions. Picture a group of hunter-gatherers moving across a late Ice Age landscape. The world is not an endless frozen wasteland, but a shifting mosaic of steppe grassland, river valleys, windswept uplands, patches of woodland, marshes, and distant ice. Herds move through the open country. Fires burn low inside temporary shelters. Children are carried across streams. Elders remember seasonal routes, and every few years the group may meet, merge with, or separate from other bands. Their population is not counted in thousands in a single settlement. It is scattered, vulnerable, and thin. A storm, an epidemic, a hunting failure, or a dangerous crossing can reduce a lineage abruptly. A successful family can leave an unusually large mark on generations to come. Within a group like this, a blood-related genetic variant might be entirely invisible. It does not change the shape of a spear point or the colour of a hide. It does not announce itself when people run, hunt, gather roots, prepare food, or sit beside the fire. A person carrying the genetic basis for an Rh-negative blood type may be as strong, vulnerable, clever, healthy, or unlucky as anyone else. In ordinary life, there is no mystical signal, no special sensation, no visible boundary between one kind of blood and another, yet the variant can be passed to children, and through them into grandchildren, crossing generations without acquiring a name. This is one of the most difficult truths about bloodline stories before modern medicine. People could not have recognised Rh status as a separate inherited trait. Ancient communities certainly understood family resemblance. They knew that children inherited features from parents and that certain illnesses, body shapes, colouring, or abilities might appear repeatedly among relatives. But negative blood does not create a recognisable clan of people who can be identified by appearance or behaviour. Its most serious consequences arise under specific circumstances, especially incompatible pregnancies or certain transfusions, and transfusion itself belonged to a very distant future. Even pregnancy losses linked to blood incompatibility would have been almost impossible to distinguish from the many other hazards of ancient childbirth and infancy. Life in prehistoric populations carries risks difficult to fully reconstruct. Infection can enter through a small wound. A difficult labour can turn fatal quickly. Infants face disease, malnutrition, cold, and injuries that leave little or no mark in the archaeological record. If a woman loses one child or several, no observer can separate immune incompatibility from fever, trauma, developmental problems, poor nutrition, or chance. The pattern that later medicine would identify remains folded inside grief. It is biologically real, but historically almost invisible. For this reason, the early history of Rh-negative blood cannot be told as the biography of a single identifiable ancestor, a first carrier standing at the entrance to a particular cave or crossing one particular mountain pass. DNA changes do not leave monuments. The evidence remains incomplete, and much of what can be said must be framed by what population genetics makes possible, rather than by a surviving eyewitness record. There may have been early carriers in more than one population. There may have been periods when the variant became more common, then rarer again, as communities expanded, separated, merged, or vanished. The present distribution of a blood type is not a photograph of its first appearance. It is the final visible surface of countless demographic events layered over tens of thousands of years. The human past is full of such events. Small populations leave Africa and encounter new climates, new foods, unfamiliar diseases, and landscapes already inhabited by other kinds of humans. Some groups spread rapidly along coasts or river systems. Some become trapped or isolated for long periods by mountains, deserts, ice, sea levels, or simply distance. Some populations contribute heavily to later peoples, others leave only fragments of their ancestry behind. Long before kingdoms draw borders on maps, the living map of humanity is being redrawn again and again by movement and disappearance. Blood traits travel with people through all of it. A variant common in one small group may arrive in a new region not because it provides an obvious advantage there, but because the people carrying it happen to become ancestors of a larger later population. This is known as a founder effect when a relatively small number of individuals establish or strongly influence a population. Their particular genetic traits can become unusually frequent among descendants. The result can look mysterious centuries or millennia later. Why does one isolated community carry a rare trait far more often than nearby populations? Sometimes the answer is not that the trait transformed their survival, but that a small ancestral group happened to carry it, and generations of relative isolation preserved and amplified the pattern. There is something almost unsettling about this. Human beings are drawn to stories in which important traits arise for important reasons. A rare inherited feature seems as though it ought to indicate a rare destiny, a hidden strength, an adaptation to some forgotten catastrophe. Occasionally, genetic traits do reveal exactly that kind of history. Variants related to disease resistance, digestion, altitude tolerance, or skin pigmentation can show the imprint of strong environmental pressures. But not every uneven genetic pattern is a survival badge. Some are footprints of population size, marriage patterns, geographic separation, and random inheritance. Nature records both necessity and accident, often in the same body. In the world before farming, before roads and written names, isolation is not an abstract concept. It is winter snow closing a pass, a coastline separated by rising waters, a community living beyond difficult terrain, a small band choosing familiar hunting grounds over uncertain travel. Even groups that are not completely separated may exchange partners, only occasionally with neighbouring populations, over generations. This is enough for certain traits to become more common locally. A feature rare across humanity can become familiar inside a small region, not because its people are a separate species or an untouched ancient race, but because their ancestry passes through a narrower channel. Later, agriculture changes the scale and rhythm of this process. Communities settle more permanently, populations grow in some regions, while disease and famine strike crowded settlements with new force. Travellers, traders, herders, and migrating farmers carry genes across landscapes more regularly. Waves of population movement do not simply replace what came before, they blend, overwhelm, preserve, and reshape it in differing proportions. A blood trait inherited from earlier hunter-gatherers may become diluted in one region, remain concentrated in another, or reappear among descendants far from the place where it was once common. What survives in the record suggests not a clean branching tree, but an intricate network of meetings, separations, bottlenecks, and returns. A bottleneck can be especially powerful. Imagine a community reduced by hunger, epidemic disease, conflict, or climatic disruption. Only a portion survives to rebuild. Whatever genetic traits happen to be carried by those survivors may become far more prominent afterward, even if the traits themselves played no role in who lived and who died. Deep time contains many such narrowed passages. Ice advances and retreats, landscapes dry or become wetter, animal herds shift their routes. Coastlines move as seas rise, human groups contract into refuges, then expand again when conditions improve. Their genes expand with them, carrying old variations into new territories. This is where the reality becomes more compelling than the fantasy, where a negative blood does not require a vanished superior race, a secret intervention, or a non-human origin to become fascinating. Its presence among living people can be approached as a remnant of ordinary humans facing extraordinary spans of time. It may have travelled through families who saw glaciers melt, who followed herds into empty valleys, who survived failed harvests, who married to newcomers, who crossed seas in small boats, who lost languages and gained others. No one carried the trait knowingly. They carried children, tools, food, injuries, memories, and genes. The blood moved because people moved. There is also a temptation to imagine a single ancient group preserving the trait in purity until the modern world finally discovered it. But human ancestry rarely cooperates with ideas of purity. Even populations with long histories of relative isolation have generally interacted with neighbours at various points. Genes pass across cultural boundaries even when languages, customs, or identities remain distinct. A community may retain a striking frequency of one trait while still carrying ancestry from multiple ancient sources. Blood does not recognise the sharp stories that later societies tell about belonging. It records reproduction, movement, survival, and contact without commentary. There is a negative trait, then enters history not as a secret emblem but as one variation among many circulating through early humanity. Its later importance does not mean early people knew what they carried. Its rarity does not mean it came from outside the human story. Its uneven distribution does not by itself reveal one lost civilization behind it. Scholars still debate the precise historical pathways by which various Rh-related genetic patterns reach their modern frequencies, especially when trying to connect living populations with prehistoric movements. Ancient DNA is beginning to offer new clues, but the farther back the search goes, the more fragmentary the evidence becomes. For the individuals living through those distant ages, all of this remains unknowable. A mother watches an infant breathe beside the fire. A hunting group crosses a valley under a hard, pale sky. A family joins another band after a season of shortage. A child inherits a silent combination of genes from parents who possess no word for it. That child grows, survives long enough to have children, and a molecular absence moves onward again. Thousands of years pass in this manner, without announcement. Eventually, descendants of these countless nameless lives build villages, towns, ports, kingdoms, laboratories, and hospitals. They learn that blood can be divided into groups. They discover that some transfusions kill while others save. They begin to see the hidden marks carried on red cells, and one of those marks, or rather its absence, receives a sign negative. Only then does an ancient inheritance become a recognized medical reality. Only then does the quiet journey through mutation. Migration, isolation, and chance begin to look backward at its own shadow and invite the question that will haunt the story from this point forward. What, exactly, is missing from Rh-negative blood, and why does that absence matter so much? By the time the ancient inheritance receives a name, the blood itself has already been carrying its secret for longer than any archive can measure. A droplet placed on a glass slide seems almost disappointingly simple, red, bright, familiar, a substance humanity has seen in wounds, childbirth, sacrifice, violence, and survival since before speech took its modern shape. Yet under the methods of 20th century medicine, that droplet becomes crowded with identities. Red blood cells are not smooth anonymous vessels drifting through the body like identical boats. Their surfaces are intricate membranes, studded with proteins and molecular structures that allow the body to distinguish its own cells from unfamiliar ones. Some of these structures differ among people. Those differences are blood group antigens, and among the most important ever discovered is the one designated by a single letter D. The familiar label Rh-negative can sound as though it describes a blood type lacking an entire biological system, or carrying some strange emptiness that separates it fundamentally from the majority of human beings. In reality, the phrase most commonly refers to one highly significant feature within a larger and more complicated family. The Rh blood group system contains many antigens, but 5 have long stood at the centre of clinical attention: D, C, E, and E. They are not floating chemicals in plasma, and they are not spiritual markers hiding inside the blood. They are parts of proteins embedded in the membrane of red blood cells. Of these, D is the one that usually determines whether a person is called Rh positive or Rh negative. If red cells carry the D antigen, the person is generally described as Rh positive. If they do not, the person is generally described as Rh negative. There is a detail here that seems minor at first and turns out to be essential. There is no ordinary opposite substance called a negative antigen. Rh negative blood is not blood bearing a rival marker known as negative. The negative sign means that D is absent, or not expressed in the expected way. In older systems of notation, scientists sometimes used a lowercase d to represent the lack of D, which could give the impression that D was itself a separate antigen. It is not. This distinction matters because much of the mystery surrounding Rh negative blood begins with the idea that it contains something unusual. In the most common forms, what makes it medically notable is not an exotic addition, but an absence, a missing surface feature that can be recognised as foreign when it appears later on donated blood cells or fetal cells. That absence is encoded not in legend but in DNA. On chromosome 1 sit 2 closely related genes called RHD and RHCE. The RHD gene ordinarily provides the instructions for making the protein associated with the D antigen. The RHCE gene contributes to the closely related C, E, and E antigens. The two genes resemble one another strongly, as though they are neighbouring variations on an ancient biological theme. The resemblance also makes the system complicated. Genes can be deleted, rearranged, altered, or combined in ways that produce weak or partial expression of Rh antigens. Not every case fits neatly into a simple positive or negative box, but in many Rh negative people of European ancestry, the central explanation is relatively direct: the RHD gene is absent, and without it the red cells do not display the D antigen in the usual form. Nothing about this absence changes the colour of the blood. It does not make the heart beat differently. It does not provide heightened senses, unusual body temperature, altered mental ability, or any of the other traits often attached to Rh negative identity in popular lore. In healthy people, ordinary Rh negative status is simply one inherited variation among the thousands carried silently through a body. A person may never know it exists until donating blood, receiving prenatal testing, undergoing surgery, or being admitted to a hospital. There is a strange humility in the fact that one of the body's most medically consequential classifications can remain invisible through childhood, adulthood, work, hunger, sleep, love, and old age, only revealing itself when blood encounters other blood. When the blood groups first began to emerge from obscurity, even scientists did not immediately see the entire structure. The discovery of the ABO system at the beginning of the 20th century transformed transfusion medicine because it explained why mixing certain human blood samples caused cells to clump and why some transfusions ended in sudden catastrophe. Yet ABO did not explain everything. Some transfusions still caused reactions despite apparently careful matching. Some pregnancies still ended in illness that medicine could observe but not properly account for. There were additional differences hidden on the red cell membrane, distinctions that the first great breakthrough had not captured. The Rh story entered view through laboratory experiments and clinical puzzles in the late 1930s and early 1940s. Researchers identified an antibody reaction connected to a red cell antigen that would eventually be called Rh, a name influenced by experiments involving rhesus monkeys. The name survived, even after it became clear that the human antigen central to the story was not simply identical to the monkey factor originally involved in the work. Science often carries these historical remnants forward a useful term, outliving the imperfect assumptions surrounding its birth. The name became familiar, but the system beneath it proved far more intricate than its short label suggested. A blood bank today can test for the presence of D with an apparent simplicity that conceals decades of investigation. A sample of red cells is brought into contact with specially prepared antibodies. If the D antigen is present, those antibodies bind to it and cause a visible reaction under controlled conditions. In ordinary language, the result becomes positive. If no detectable D reaction occurs, the sample is classified as negative, though difficult cases may require more refined testing. What looks like a plus or minus sign printed on a donor card is therefore the outcome of a molecular recognition event and antibody finding, or failing to find, a particular target on the surface of red cells. This is the same basic principle that gives the immune system its power. The body is filled with recognition systems. It learns the molecular shapes that belong to it, and it reacts when it encounters certain unfamiliar shapes associated with danger or foreign cells. That capacity is indispensable. Without it, infection would move through the body almost unchecked. But recognition also has consequences when one person's red blood cells enter another person's circulation. A D-negative individual has red cells that do not display the D antigen. If D-positive red cells arrive through transfusion, or through the limited exchange of cells that can occur between fetus and mother, the immune system may interpret D as an outsider, and may then build antibodies designed to recognize D if it appears again. The D antigen has particular medical importance, because it is highly capable of provoking this immune response in someone who lacks it. It is not the only blood group antigen that can create trouble. Medicine now recognizes many such possibilities. But after ABO, D has become one of the central concerns in blood matching because antibodies against it can produce serious consequences. The body does not understand that a transfusion has been given to save a life, or that fetal cells carry a mixture of parental inheritance. It recognizes molecular difference. The immune response that protects against threats in one context can become dangerous in another. To describe this as an immune mistake is understandable, though not entirely precise. From the body's perspective, it is doing what it has evolved to do, identifying unfamiliar structures and preparing against them. The tragedy lies in circumstance. Medicine now places cells from one human bloodstream into another. Pregnancy naturally brings genetically distinct life into intimate biological contact with the mother. In these circumstances, a difference that is harmless in ordinary daily existence can suddenly matter. The same Rh-negative person who walks through decades without symptoms may, under the right conditions, produce antibodies that shape future transfusions or pregnancies. No single explanation fully accounts for every individual immune response, because not every exposure leads to antibodies in exactly the same way, and the Rh system includes variants that complicate neat classification. But the central danger is well established. The complexity reaches beyond simple absence. Some people possess forms of the Rhd gene that cause their red cells to express the D antigen weakly. Others express only parts of the usual D antigen, a condition often referred to as partial D. To routine eyes, these cases can look confusing. A person may appear positive in one test, uncertain in another, or be vulnerable to producing antibodies against portions of D that their own cells do not carry. These variants remind scientists that human genetics is not designed to fit the categories printed on hospital forms. Positive and negative are clinically useful labels, but beneath them lies a spectrum of genetic structures shaped by mutation and inheritance. There are still rarer conditions, so unusual that they have acquired their own mythology. A person described as Rh null lacks all of the major Rh antigens on the red blood cells. Not only D, this is not the same as ordinary Rh negative blood. It is exceptionally rare and medically significant, both because such patients may have difficulty finding compatible blood, and because the Rh proteins contribute to the stability of the red cell membrane. Null blood has sometimes been called golden blood, a phrase that invites visions of biological nobility or extraordinary power. Its true value is more practical and more poignant for certain extraordinarily rare patients. A matching donation may be almost impossible to find. The rarity makes the blood precious in a blood bank, not magical in the person who carries it. This distinction is often lost as stories pass from medical reality into internet folklore. Rh-negative becomes confused with Rh-null. Medical rarity becomes spiritual distinction. Complex genetics becomes proof that science is hiding something. Yet the real cellular architecture is richer than these simplifications. Rh proteins do not sit uselessly on the cell, like labels pasted there solely to cause trouble during transfusion. They form part of a membrane complex associated with the red cell's physical structure, and researchers explored their possible roles in transporting certain substances across the membrane. Their full biological story is not reduced to one settled sentence. Scientists understand their clinical importance far better than every aspect of their original physiological function. Scholars still debate parts of that deeper function, and the evolution of the Rh system across human populations remains a field where additional genetic evidence can sharpen the picture. That incompleteness is not concealment. It is the ordinary condition of serious science. A blood group system can be understood well enough to prevent deaths, while still containing unanswered questions about molecular function, genetic history, and the precise pathways by which variants rose or fell among ancestral populations. Medicine does not need a final theory of every ancient mutation to know that anti-D antibodies can be dangerous. A laboratory does not need to reconstruct the entire journey of a family line across prehistoric Europe to determine whether a donor's cells carried its. What remains unknown is not proof that the known part is fraudulent. It is proof that human biology is old, layered, and difficult to read completely. There is, however, something undeniably evocative about the D antigen once its nature becomes clear. It is a feature so small that millions could fit within the spaces of ordinary imagination, yet its presence or absence has consequences reaching from invisible genetic inheritance into the most vulnerable moments of life. It can matter when a surgeon calls for blood, when a pregnant patient is tested early in care, when a donor's rare compatibility is entered into a registry. When an infant's red cells are threatened by antibodies formed before birth, it is neither a sign of chosen ancestry nor a meaningless laboratory curiosity. It is an ordinary molecular difference, with an extraordinary ability to reveal the intimate tension between one human body and another. For most of history, that tension remained unexplained. A mother could carry life within her while an invisible immune memory gathered strength against a trait inherited from the child's father. A later infant could become dangerously ill, and no one around the bed would know that the cause lay on the surface of red blood cells, in the presence of a single antigen and the antibodies built against it. This was the darker edge of the small absence carried forward from deep time. The negative sign did not announce disease in the person who bore it. It waited until two blood inheritances met, most tragically, where human beings expected only continuation, birth, and family. The danger is most profound because pregnancy is not normally a struggle between hostile bodies. It is an extraordinary negotiation. A fetus grows within the mother while carrying a genetic inheritance that is partly her own and partly derived from the father. The placenta does not simply separate them like a wall. It is a living interface, allowing oxygen and nutrients to pass toward the fetus, carrying waste away, regulating hormones, and managing a delicate immunological balance that permits one distinct human life to develop inside another. Most of the time, differences between maternal and fetal blood remain manageable. But when an Rh-negative mother carries an Rh-positive fetus, a few red blood cells crossing that boundary can sometimes begin a chain of events whose consequences may not appear until much later. The fetus can inherit the D antigen from an Rh-positive father. Inside the fetus, this is entirely normal. Its red cells carry D because its genes instruct them to do so. The mother, if she is Rh-negative, lacks that antigen on her own red cells. During much of pregnancy, the maternal and fetal circulations remain largely separate, but separation is not absolute. Tiny quantities of fetal blood can enter the mother's bloodstream, particularly late in pregnancy or during delivery after miscarriage. Bleeding, abdominal trauma, certain prenatal procedures, or other moments when the placental boundary is disturbed, a handful of fetal D-positive red cells may be enough for the mother's immune system to encounter an antigen it has never seen as part of itself. At first, nothing visible may happen. There is no sensation by which a mother can feel that sensitization has begun. No fever announces it. No change in fetal movement necessarily reveals it. The immune system processes the unfamiliar D antigen and, in some women, gradually forms anti-D antibodies. This first immune response may come too slowly to cause serious harm to the pregnancy in which exposure initially occurs, especially when most of the fetal blood enters the maternal circulation around delivery. That is why, historically, a first Rh-positive baby born to an Rh-negative mother was often healthy, but the mother's immune system now possessed a memory. The next time it encountered D-positive fetal red cells, it could respond faster and more forcefully. The cruelty of this pattern once seemed almost deliberately obscure. A family might welcome a healthy first child, then face increasing tragedy in later pregnancies. A second baby might be born jaundiced and weak, another might become dangerously swollen before birth, another might not survive at all. Without a way to identify Rh incompatibility, the sequence looked irrational. There were no contaminated wells to blame, no visible injury, no obvious inherited deformity. The mother herself might remain physically healthy, the father might be healthy, the first child might be thriving, yet later infants could be attacked before they had taken a breath. Their red blood cells destroyed by antibodies crossing the placenta from a mother whose immune system was reacting to an antigen inherited innocently from the father. These antibodies are especially dangerous because of their type. After sensitization, maternal anti-D antibodies can include immunoglobulin G, or IgG, a class of antibody capable of crossing the placenta. Once across, they attach to the D-positive red cells of the fetus. The fetal body then removes those marked cells from circulation more rapidly than it can replace them. At the mild end, the newborn may become anemic or jaundiced because the breakdown of red blood cells releases bilirubin. At the severe end, the fetus becomes profoundly anemic while still in the womb. The heart must work harder to deliver enough oxygen. Fluid can accumulate in tissues and body cavities, creating a condition known as hydrops fetalis. Without intervention, the result can be heart failure, brain injury, stillbirth, or death soon after delivery. Long before this chain was understood, its signs were already present in human life. The condition left traces not in grand monuments or medical texts capable of explaining it, but in nurseries, birth rooms, burial grounds, and family memory. Newborn jaundice could be seen, though its underlying causes varied. Severe swelling in stillborn or dying infants could be observed, though not properly interpreted. Midwives and families may have noticed that some women lost more infants than others, or that losses appeared after an earlier successful birth. But pregnancy and infancy were vulnerable for countless reasons. Infection, malnutrition, prematurity, birth trauma, genetic disorders, and maternal illness all overlapped in the same human suffering. A specific immune conflict hidden inside blood was effectively indistinguishable from the many other ways life could fail before modern diagnosis. Even in the early modern hospital, the puzzle resisted explanation. Physicians could observe unusually severe jaundice in newborns, anemia, or infants born with widespread swelling. The conditions were given clinical names, described carefully, and feared, yet separate appearances of the same disease did not immediately seem connected. A newborn with brain damage from extreme jaundice, another with anemia, and a stillborn infant with hydrops might represent stages of one process. But only once blood group science advanced could that unity begin to appear. In the first half of the 20th century, the pieces finally aligned maternal antibodies, fetal red cell destruction, and the Rh factor were recognized as part of a single, devastating mechanism. The condition became known as hemolytic disease of the fetus and newborn. The name is clinical, almost restrained, for something that had torn through family histories without warning. Hemolysis means destruction of red blood cells. In this case, it was happening not because an infant's blood was malformed in some obvious way, but because those cells carried an antigen the mother's sensitized immune system had learned to attack. Once that mechanism was understood, medicine could begin to identify pregnancies at risk. Blood typing could determine whether a mother was Rh negative. Antibody screening could reveal whether she had already formed anti-D. Doctors could observe infants and, later, fetuses for evidence of anemia. The invisible enemy at last became visible through testing. But recognition did not immediately erase the danger. Before reliable prevention, physicians faced pregnancies in which antibodies were already present and fetal red cells were being destroyed. Treatment could be desperate and astonishingly bold. Severely affected newborns might undergo exchange transfusions, in which portions of the infant's blood were gradually replaced to reduce antibodies and bilirubin while restoring healthier red cells. Later advances made it possible, in some cases, to transfuse an anemic fetus before birth. Such interventions could rescue lives, but they came after the conflict had already begun. They required expertise, timing, and the acceptance of considerable risk. Medicine had learned how to fight the damage, but the greater achievement would be preventing the immune memory from forming at all. That breakthrough arrived through one of the more elegant reversals in medical history. Instead of waiting for an Rh-negative mother's immune system to recognize D-positive fetal cells and begin building its own long-lived defense, doctors could give her prepared anti-D immunoglobulin at carefully chosen times. These supplied antibodies bind to D-positive fetal red cells that have entered her circulation and help clear them before her immune system becomes sensitized in the dangerous, lasting way. It seems paradoxical at first to prevent harmful antibodies by giving antibodies, yet this is precisely the beauty of the method. The administered anti-D acts quickly and temporarily, removing the trigger before the maternal immune system develops the memory that could threaten later pregnancies. The prevention is commonly offered during pregnancy to Rh-negative women who are not already sensitized, and again after delivery if the newborn is confirmed to be Rh-positive. It may also be needed after events that could allow fetal blood into the maternal circulation, such as miscarriage, certain bleeding episodes, abdominal injury, or procedures involving the pregnancy. The exact protocols vary by country and clinical circumstance, but the principle is constant. It is not a treatment for a strained bloodline. It is a carefully timed intervention that prevents a normal immune system from learning a target it must never be allowed to attack in a future fetus. The effect has been immense. In places where prenatal screening and anti-D prophylaxis are reliably available, the once common pattern of Rh-related infant sickness and death has been dramatically reduced. A risk that once haunted successive pregnancies can now often be anticipated early and prevented with routine care. The simplicity of the modern appointment can hide the scale of what has been achieved. A blood sample is taken, a chart is marked, an injection is given at the right stage. Perhaps another afterbirth. A mother leaves the clinic and continues her pregnancy, often unaware that the ordinary rhythm of her care rests on generations of children who could not be saved before this mechanism was understood. Not all cases vanish with prevention. Some women are already sensitized before a pregnancy is recognized, perhaps through an earlier pregnancy, a miscarriage, a previous transfusion, or an unprotected exposure. Anti-D prophylaxis cannot erase established immune memory once significant antibodies are formed. Other blood group antibodies beyond anti-D can also cause fetal disease and require monitoring. In parts of the world where access to testing, injections, blood products, specialist surveillance or emergency obstetric care is limited, preventable illness and loss still occur. What seems like a solved problem from the vantage point of a well-equipped hospital is not equally solved everywhere. The evidence remains incomplete in some settings because medical records, testing access, and treatment availability vary, but there is no doubt that prevention has transformed the course of Rh incompatibility where it can be delivered. This history also explains why Rh-negative blood acquired a reputation far larger than its everyday biological effect. For the individual living without pregnancy or transfusion concerns, being Rh-negative usually changes nothing perceptible. Yet within reproductive history, the trait could appear to mark families with an invisible curse, later children becoming ill, mothers grieving repeatedly, physicians unable for centuries to explain why. Such patterns have always attracted supernatural interpretation, blame, and fear. In older societies, repeated infant loss might be attributed to fate, wrongdoing, contamination, divine anger, or the supposed weakness of a mother. The scientific explanation, when it finally arrived, was both colder and more merciful. The mother was not cursed, her blood was not corrupted, her body was defending itself according to rules that, in one heartbreaking circumstance, placed the fetus in danger. There is an additional detail often missed in simplified accounts. An Rh-negative woman does not automatically face a threatened pregnancy. If the fetus is also Rh-negative, there is no D antigen for her immune system to attack. Even if the fetus is Rh-positive, modern preventive care usually prevents sensitization when provided appropriately, and even without prevention, not every exposure results in the same degree of antibody development or disease. Biology is not a script that unfolds identically in every body. Risk depends on inheritance, prior exposure, immune response, clinical care, and timing. The popular idea that Rh-negative mothers are naturally incompatible with ordinary human reproduction is both wrong and needlessly alarming. Millions of Rh-negative women have healthy pregnancies and healthy children. Yet the older danger was real. It rests at the centre of the art story because it demonstrates how a trait invisible in normal life can acquire terrifying importance when generations meet. Blood is inheritance, but pregnancy is inheritance placed in contact maternal cells, fetal cells, paternal contribution, immune memory, and the unpredictable boundary between protection and harm. The medical solution did not arrive by uncovering a secret ancient origin. It arrived by patiently identifying antibodies, understanding their movement across the placenta, developing reliable testing, and discovering how to prevent the immune system from remembering D as an enemy. In a modern maternity ward, the aftermath of this discovery is almost quiet. An Rh-negative mother holds a newborn whose red cells may carry the D antigen she lacks. The child is warm, restless, alive, with no awareness of the molecular difference that once might have placed future siblings in danger. A nurse checks records. A test confirms the infant's blood type. Anti-D immunoglobulin is arranged, not with drama, but with the calm efficiency of a danger made familiar and manageable. It is one of medicine's most remarkable forms of progress, the prevention of a tragedy so complete that later generations can forget how often it once occurred. But once a trait has entered human imagination through birth, death, and family inheritance, facts alone do not easily dissolve its aura. The hospital can explain the immune mechanism, the clinic can prevent the worst consequence. Still, another question gathers at the edge of the story. If Rh-negative blood is inherited, and if it's far more common among certain populations than others, where do those patterns come from? The answer does not lie in the delivery room alone. It begins to appear on maps, in remote valleys, along ancient migration routes, and within communities whose blood frequencies seem, at first glance, to preserve something unusually old. The map begins with a simple observation: the negative sign is not distributed evenly across humanity. In one maternity clinic, one donor centre, one isolated valley, or one crowded coastal city, Rh-negative blood may appear with very different frequency from another place thousands of miles away. That unevenness is real, is one of the reasons the story has endured beyond the hospital. Once people learn that a trait affecting transfusion and pregnancy is noticeably more common in some ancestral populations than in others, the imagination begins arranging the pattern into secret routes and forgotten tribes. But maps of blood do not speak so easily. They are not treasure charts, they are palimpsests, written and rewritten by movement, marriage, isolation, population decline, expansion, disease, and chance. Stand far enough back from the world, and the broad pattern is striking. Among people of much European ancestry, roughly one person in 6 or 7 may be Rhd negative. In many populations with ancestry in East Asia, the proportion is dramatically lower, often well below 1%. Across African populations, the picture is more varied and molecularly more complex, but Rhd negative blood is generally less frequent than in much of Europe. Between these broad regions lie countless local histories, each complicating any easy summary communities shaped by trade routes, mountain isolation, colonial displacement, pastoral migration, intermarriage, forced movement, famine, and the uneven accidents by which some family lines expand while others fade. A modern blood bank sees these differences as practical reality. In a region where Rh-negative patients and donors are relatively common, hospitals can maintain supplies with one kind of planning. In a place where Rh-negative blood is exceptionally uncommon, finding compatible units for an Rh-negative patient can be far more difficult, especially when that patient also has antibodies against additional red cell antigens. The difference is not theoretical. It determines how donor registries are built, how rare units are stored, how emergency policies are written, and sometimes how urgently hospitals must search across regions or national borders for a suitable donation. A genetic frequency shaped in the distant past still exerts pressure on shelves of refrigerated blood today. To anyone searching for the origins of Rh-negative blood, Europe draws attention first. The negative phenotype appears there at relatively high levels compared with many other regions of the world. That fact has been repeated so often in popular accounts that it is frequently treated as if it points directly to a single ancestral source. But Europe has never been a sealed genetic container. Its population history is a long sequence of arrivals, contractions, expansions, and mixtures. Ice Age hunter-gatherers occupied refuges when cold made northern landscapes difficult or impossible to inhabit, then spread again as climates changed. Later, farming communities moved in from Anatolia and neighbouring regions, bringing new ways of life and new ancestry. Still later, pastoral groups from the Eurasian steppe contributed heavily to many European populations. After that came migrations, conquests, trading networks, slave routes, empires, epidemics, religious movements, and ordinary marriages that rarely enter historical records but matter greatly in genetic history. The relatively high European frequency of Rh-negative blood is therefore not a simple tag pinned to one vanished people. It may reflect combinations of founder effects, ancient population structure, drift, and later mixture. It may also conceal different local histories under the appearance of a single shared trait. A person in Ireland, a person in Iberia, and a person in Central Europe can all be Rh-negative without their negative status, having arrived through one neat and exclusive ancestral line. The blood test records where the D is expressed on the red cells. It does not, by itself, identify the entire path by which that absence was inherited. That caution becomes even more necessary when looking beyond Europe. In many people of European descent who are Rhd negative, the explanation often involves deletion of the Rhd gene. But in African populations, a person may test as Rhd negative for more than one genetic reason. Some carry a deletion, others may carry altered forms of Rhd that do not produce ordinary D antigen expression, including pseudogenes or hybrid arrangements formed from the close relationship between Rhd and Rhce. The same clinical label, negative, can therefore conceal distinct molecular backgrounds. Two people may appear identical on the basic blood typing result while carrying different genetic histories beneath it. This is a surprisingly important detail, because it undermines one of the most common assumptions in bloodline mythology, that all Rh-negative people belong to a single hidden lineage. The surface result may be the same, but the genetic route to that result need not be identical across populations. Biology is capable of arriving at similar outward outcomes through different inherited pathways. A missing antigen on a red blood cell is not automatically a shared passport from one ancient tribe. It may be a family of related stories, some overlapping, some independent, some altered by centuries of population contact that no written record fully preserves. The low frequency of Rh-negative blood across much of East Asia offers another kind of lesson. In many East Asian donor populations, ordinary Rhd-negative blood is extremely rare. That rarity creates serious logistical challenges for patients who need compatible transfusions, and it has led researchers and blood services to pay close attention to unusual variants. In some individuals who seem Rhd negative under routine testing, minute traces of D may still be present in a form called DEL, sometimes detectable only through specialized methods. Again, the positive-negative division that appears so absolute on an identification card becomes more nuanced when examined closely. A trait can be rare, and its rare appearances can still consist of several different molecular forms. Popular mystery narratives usually ignore these details because complexity is less satisfying than a hidden origin story. A map with high frequencies here and low frequencies there is tempting to interpret as evidence of ancient difference in a grand, dramatic sense. But genetic maps are often shaped by processes that are both more ordinary and more powerful than legend. A small founding community carrying a trait by chance, centuries of marriage mostly within a region, a sudden population decline, the expansion of descendants from a limited number of survivors, later contact introducing other variants, and the gradual blurring or preservation of earlier patterns. There is no requirement that a trait be useful, supernatural, or imposed from outside humanity in order for it to become common in one population and scarce in another. Consider a remote mountain valley. For much of its history, the population might be connected to the wider world, but not evenly. Merchants pass through occasionally. Armies may arrive once in a generation. Marriage across the mountains occurs, though less frequently than marriage among nearby villages. If a blood variant is already relatively common among the valley's founders, it can remain conspicuous for centuries, not because the inhabitants possess hidden abilities, not because their ancestors were untouched by ordinary humanity, simply because geography narrows the paths through which genes enter and leave. A mountain does not need to create a trait in order to preserve its frequency. Islands can do the same, so can deserts, forests, language boundaries, social customs, religious endogamy, or patterns of movement defined by livestock and seasonal grazing. Human beings do not reproduce at random across the planet. For most of history, Most people formed families within a surprisingly limited radius of where they were born. Their blood traits remained local, not because the wider world was inaccessible in every sense, but because ordinary life is powerful farms, kinship. Trusted neighbours, familiar roots, obligations to land, burial places, and the enormous difficulty of leaving one's known world behind. A blood map therefore contains cultural history as well as biological inheritance. It preserves the echoes of whom people met and married, whom they avoided, which communities endured, and which were absorbed into others. Yet it must be read with restraint. A higher frequency of Rh-negative blood does not make a population older than its neighbours in some simple sense. All living populations have ancient ancestry. All have undergone movement and mixture. No community alive today is a frozen sample of Paleolithic humanity sealed away from time. Some groups retain distinctive genetic patterns, certainly. Some preserve unusually strong signals of isolation or founder effects. But distinction is not purity, and rarity is not rank. The world's blood frequencies also reveal how deceptive modern political borders can be. The boundaries printed on a map are recent compared with the movements that shaped human genetics. A national percentage can blur major regional differences within the same country. Coastal populations may differ from mountain populations. Communities speaking different languages may preserve different histories. Cities may gather ancestry from distant regions that rural areas do not experience to the same extent. When online lists rank countries by Rh-negative percentages, they often flatten the very complexity that makes the subject scientifically interesting. Blood distributions follow ancestral history more than flags. Even broad racial labels, familiar in modern conversation, are blunt instruments when applied to a system as complicated as rage. Africa contains the greatest human genetic diversity on Earth, and no single African frequency can capture its populations. Europe holds local variation beneath its generally higher levels of Rhd negativity. Asia encompasses immense differences in language, geography, and ancestry. The Americas contain indigenous ancestry alongside complex histories of European, African, and Asian migration and forced displacement. Any claim that root-negative blood cleanly defines one race, one lineage, or one civilization begins by simplifying humanity until the evidence can no longer be seen clearly. And yet the distribution still demands explanation. Why should the D-negative phenotype have reached relatively high frequency in parts of Europe at all, given the historical danger it could pose in pregnancies before anti-D prevention existed? A trait that could contribute to fatal hemolytic disease might seem unlikely to persist at notable levels. This apparent contradiction has encouraged theories ranging from hidden reproductive advantages to protection from diseases, parasites, or unknown ancient pressures. Some researchers have explored whether Rh variation might interact with health or infection in subtle ways. Others emphasize demographic forces such as drift and founder effects. No single explanation fully accounts for the global distribution, and the evolutionary history of the Rh system remains more complicated than any one proposal. That unresolved question should not be exaggerated into evidence of suppression. Evolution does not always remove a trait simply because it can be harmful in a particular circumstance. The danger of Rh incompatibility depends on several conditions aligning an Rh-negative mother, an Rh-positive fetus, fetal blood exposure sufficient to sensitize her, the survival and continuation of later pregnancies, and no modern preventive care. The trait can persist because its costs are conditional rather than universal, because not every carrier is a mother, not every pregnancy involves an Rh-positive fetus, not every exposure produces serious sensitization, and population history can preserve variance despite disadvantages. Human genomes contain many features whose survival reflects complicated trade-offs, limited costs, or sheer demographic momentum rather than obvious perfection. For families and blood services, these debates are not abstractions. A woman who learns she is Rh-negative during prenatal care may discover that the trait is more common among relatives than she realized. A donor center may contact certain Rh-negative donors repeatedly because their blood is urgently useful. A patient with a rare combination of antibodies may require a search through registries that span countries and ancestries. In each case, the map becomes personal. Lines of inheritance that wandered through ancient populations arrive in one body, in one hospital room, in one moment when compatible blood must be found. And among all the regions that have captured attention, one population has become almost inseparable from Rh-negative speculation, living on either side of the Western Pyrenees, speaking a language that does not belong to the Indo-European family surrounding it, and long regarded as culturally distinctive. The Basque people have repeatedly been drawn into stories of ancient survival and unusual blood. The relatively high Rh-negative frequency became, for many observers, more than a medical statistic. It became a clue, perhaps even the clue, to a supposedly isolated bloodline reaching back before the arrival of later European peoples. The appeal of that idea is easy to see. Mountains, an enigmatic language, old customs, and rare blood seem to align into a perfect mystery. But the closer one looks, the more subtle and more human the Basque story becomes. At the western end of the Pyrenees, where green mountain slopes descend toward the Bay of Biscay and narrow valleys turn geography into a kind of shelter, lives a people who have been made to carry more mystery than almost any other population in Europe. Their homeland crosses the modern border between northern Spain and southwestern France. Their language, Euskara, stands apart from the Indo-European languages that surround it. Spanish, French, English, German, Celtic languages. Slavic languages, Greek, Persian, and many languages of northern India all belong, however distantly, to the vast Indo-European family. Euskara does not. It is not a dialect left behind by Rome, nor a surviving branch of Celtic speech, nor a language imported by some clearly documented medieval migration. Its deeper origins remain uncertain, and for generations, that linguistic isolation seemed to align irresistibly with another unusual observation. The Basque population was reported to have one of the highest frequencies of Rh-negative blood in Europe. Here, at last, the popular story appears to find its ideal setting: a mountain people, a language unlike its neighbours, blood frequencies that seem unusually distinct, a region at the edge of Atlantic Europe where caves, old stone monuments, shepherding paths, and rain-darkened ridges seem capable of preserving an inheritance from before history began. It is not difficult to see why writers once imagined the Basques as direct survivors of Europe's most ancient inhabitants, a people whose ancestors withdrew into the Pyrenean landscape, while later populations flowed around them. In the most exaggerated versions, the high occurrence of Rh-negative blood became proof that they preserved an untouched lineage, older and somehow separate from the rest of Europe. The real story is more restrained, but it does not make the Basques less remarkable. It makes their survival more believable. For much of the 20th century, researchers studying blood groups noticed unusual frequencies among Basque communities, including in the ABO, Rh, and other blood group systems. This was before scientists could read large stretches of the human genome, before ancient DNA could be retrieved from old bones and compared with living populations. Blood group frequencies served as early markers of population difference. If a community showed unusual proportions of certain inherited blood traits, researchers could reasonably suspect that its demographic history differed from that of surrounding communities. The Basques did show distinctions, and a negative blood became the most famous of them. Because the trait was already medically significant and publicly familiar, it escaped the laboratory and entered the imagination. The older interpretation often treated the Basques as a remnant of Paleolithic Europe descendants of hunter-gatherers, who had survived the arrival of Neolithic farmers and later Indo-European-speaking migrants with unusually little change. Their language appeared to strengthen the case, if Euskara was the last surviving language of pre-Indo-European Western Europe. Perhaps Basque genes also preserved a similarly ancient isolation. It was an elegant idea, almost cinematic in its simplicity. And old people hold out among mountain valleys. Their language survives. Their rare blood preserves a biological signature of a vanished continent. But elegance is not proof, and modern genetics has forced this picture to become considerably more complicated. The first correction is that the Basques are not genetic outsiders to the rest of Iberia or Western Europe. Their ancestry is formed from many of the same broad ancient ingredients seen in neighbouring populations. Long before a Basque cultural identity can be traced, the Iberian Peninsula had already undergone profound human transformations. Hunter-gatherers lived there after the Ice Age, farming peoples arrived and mixed with local inhabitants. Later, ancestry linked to pastoral populations from the Eurasian steppe spread into Iberia during the Bronze Age. Earlier theories sometimes imagined the Basque region as largely untouched by this steppe-related movement, as though its people had escaped the demographic changes affecting much of Europe. Ancient DNA now indicates otherwise. The ancestors of present-day Basques were not sealed off before these major transformations. They participated in the formation of Bronze and Iron Age Iberian populations. This changes the timing of the mystery. The Basques do not appear to be a perfectly preserved pocket of Europe before farming, nor a population untouched since the age of Ice Age cave artists. Rather, genomic evidence suggests that their distinctiveness becomes especially clear after many of the great prehistoric mixtures had already occurred. By the Iron Age, roughly 3,000 years ago, communities ancestral to modern Basques resemble other Iberian populations of that period. Afterward, however, while many other parts of Iberia experienced additional gene flow associated with later Mediterranean, North African, Roman period, medieval, and other historical contacts, the Basque region shows far greater continuity and reduced outside mixture. The difference is not that Basque ancestors came from an entirely separate human source. It is that, after sharing in much of Iberia's earlier formation, their descendants remained comparatively less altered by later population movements. This is a far subtler form of survival. It is not purity preserved from the dawn of Europe. It is continuity maintained across turbulent historical centuries. While empires occupied roads and ports, while soldiers, merchants, administrators, settlers, enslaved people, and refugees carried ancestry across the peninsula, the valleys and communities of the Basque country remained less penetrated by some of those later genetic currents. Geography helped. But mountains alone are never a complete explanation. The Western Pyrenees are not an impenetrable wall. People crossed them, traded across them, fought through them, and married across them. Isolation is rarely absolute. It is more often a matter of frequently fewer outside marriages, stronger local networks, more persistent community boundaries, and enough generations for small differences to deepen. Language may have been one of the strongest boundaries of all. Euskara was not merely a relic spoken in private, while all other aspects of life blended evenly with neighbouring regions. Language creates communities of trust, marriage, trade, belonging, memory, and identity. A person is often more likely to form a household where speech, custom, and kinship already align. Researchers studying modern Basque genetic structure have found that areas where Euskara endured most strongly can also show patterns of greater genetic continuity, while internal dialect boundaries may reflect subtle local separation as well. A language does not prevent contact like a fortress wall, but over centuries it can make some contacts more common and others less so. The surprising detail is that the most powerful barrier shaping a population may not be the ridgeline visible on a map, but the words spoken across a family table. Against this background, the high frequency of Rh-negative blood becomes understandable without becoming trivial. If Rh-negative variants were already present at notable levels in ancestral populations of the region, then centuries of relative continuity and reduced later gene flow could help preserve those frequencies. Genetic drift may also have played a role, especially within smaller or more locally structured communities. A rare trait can become more common or remain unusually common, without conferring special strength and without indicating descent from an entirely separate race of people. The Basque frequency is a clue to demographic history, but it cannot by itself identify one moment of origin, one founding ancestor, or one ancient population responsible for all Rh-negative blood elsewhere. The blood itself has also proved more complicated than the early legends allowed. Research examining the Rh genetic region in Basques does not reveal a biological system outside normal human variation. Basque Rh-negative patterns fit within the wider genetic histories of Western Eurasian populations, even if local frequencies are distinctive. There is no hidden Rh gene that transforms the Basques into a separate category of humanity. No markers showing that their blood came from an unknown non-human source, and no support for claims that the trait confers unusual powers. Their blood is medically important for exactly the same reasons Rh-negative blood matters anywhere: transfusion compatibility, pregnancy care, inheritance, and the practical value of donor availability. Yet one can understand how the exaggerated story took hold. For people searching for mystery, the Basques seem almost designed to be misunderstood. Their language is genuinely exceptional. Its relationship to ancient languages elsewhere in Europe remains uncertain because those languages are mostly gone, leaving too little evidence for secure reconstruction. Their population history does show unusual continuity since the Iron Age. Their Rh-negative frequency did attract scientific attention. Each part of the puzzle is real. The error appears when these pieces are forced to prove more than they can an unbroken Paleolithic bloodline, a separate origin, or evidence of something deliberately concealed by science. There is also a deeper danger in such claims. To call a population pure or biologically separate may sound admiring in a mystery narrative, but history shows how easily ideas of blood distinction become burdens placed upon real people. The Basques are not specimens preserved behind glass. They are communities whose identities have been shaped by language, land, political conflict, farming and fishing, urban life, migration, and the ordinary complexity of families. Their genetic history is significant because it illuminates the human past, not because it ranks them above or outside other people. A high frequency of one blood phenotype cannot carry the full weight of a culture. Within the valleys themselves, as negative blood would have passed quietly through families just as it did elsewhere. A shepherd moving sheep through wet upland pasture, a fisher working Atlantic waters, a mother preparing food in a stone farmhouse, a child learning the words of a language spoken nowhere else in quite the same way none would feel the RHD antigen absent from their red cells. Before blood testing, no family could point to it as a visible inheritance. If Rh incompatibility contributed to infant losses, it would have remained buried among the universal dangers of birth and early life. Only the modern laboratory could take this invisible feature and place it beside linguistic difference and geographic continuity, inviting larger interpretations. What survives in the record suggests that the Basque story is not the tale of a hidden ancient race revealed by blood, but of a population whose distinctiveness grew through historical continuity, reduced mixture after the Iron Age, and the durability of language and community boundaries. Their ancestors were involved in the same large prehistoric transformations that reshaped Iberia. They were not untouched by farming populations or Bronze Age migrations, but later. While many surrounding populations absorbed further layers of ancestry, Basque communities retained more of an earlier regional profile. Rh-negative blood became one visible thread in that continuity, not the cause of it, and not the proof of an impossible isolation. The mountains therefore preserve a mystery, but not the one often advertised. The mystery is not why the Basques are unlike humanity. They are not. The mystery is how cultures and populations remain recognizably themselves, while history moves through and around them, how a language survives when relatives of that language vanish, how small patterns in blood can persist across wars, borders, empires, and migrations, how continuity can look from a distance, like an untouched beginning when it is really the result of countless human choices repeated across centuries. Beyond the Pyrenean valleys, the wider European landscape carries the same question on a far larger scale. The Basque case points backward, toward the climatic upheavals and migrations that formed the populations of Ice Age and post-Ice Age Europe, toward refugees where people endured when cold narrowed the habitable world, and toward later expansions that carried inherited traits across an opening continent. The negative sign did not belong to one mountain people alone. Its trail leads farther back, into landscapes where ice, sea, steppe, forest, and survival continually remade the human map. Long before the valleys of the Pyrenees became associated with a surviving language or an unusual blood frequency, Europe itself was a place of repeated disappearance and return. The continent recognised on modern maps did not exist in anything like its present human form. Vast ice sheets pressed southward across the north. Alpine glaciers reached down through mountain systems. Sea levels fell as water became locked in ice, exposing coastlines now drowned beneath the Atlantic, the North Sea. And the Mediterranean approaches. Forests withdrew, returned, and withdrew again. Herds shifted across grasslands where cities would one day stand. Human communities followed food, avoided danger, found shelter, and endured cold so persistent that it reordered the living world around them. Any rare inherited trait present among those communities would be carried not through stable nations or named ethnic groups, but through fragments of humanity repeatedly pushed into refuges and then released across an altered landscape. During the most severe phases of the last ice age, especially near the last glacial maximum more than 20,000 years ago, much of northern Europe was difficult or impossible for sustained human occupation. The habitable zones contracted southward and westward into regions where climate, shelter, animal resources, and access to water still made survival possible. Parts of the Iberian Peninsula, southern France, Italy, the Balkans, and areas farther east served at different times as refuges for plants, animals, and humans. The word refuge may suggest safety, but prehistoric refuge was never comfortable. It meant places less lethal than those abandoned, not places free from hunger, cold, disease, or loss. In rock shelters and cave mouths, along river corridors and coastal plains now submerged, small populations made tools, processed animal hides, maintained fires, exchanged ornaments and materials across distances that reveal networks of contact even in harsh times. These were not faceless survivors wandering without culture. Their world was full of memory and skill. They knew the behaviour of reindeer, horse, ibex, bison, and red deer. They selected flint from particular sources and shaped it into blades and points. They made needles from bone, stitched clothing, used pigments, and left behind carved objects and painted caves whose animals still seemed to move in the flickering imagined light of a fire. Some of the greatest Ice Age art survives in southwestern Europe, including regions near the later Basque homeland, which encouraged older scholars to connect modern populations directly with the artists of the caves. But paintings cannot assign blood groups to the people who made them. They reveal minds, animals, symbols, and extraordinary continuity of human attention. They do not identify an Rh negative ancestral tribe. What Ice Age survival does provide is a powerful framework for understanding how a rare genetic variant can become unevenly distributed. When a population contracts into smaller groups, it passes through a bottleneck. Thousands of individuals scattered across broad territories may be reduced to fewer surviving communities. Within those communities, the genes carried by particular families become disproportionately important. A variant that was uncommon before the contraction can, by chance, become more frequent among survivors. When climate later improves and descendants expand into newly habitable territory, they do not carry an average sample of all earlier populations. They carry the particular inheritance of those who endured in the refuges. The expanding people become, genetically, a widening echo of a narrowed past. Imagine a sequence extending beyond a single lifetime. A cold period intensifies, hunting grounds fail farther north, small bands retreat over generations toward areas with more dependable game, shelter, and plant foods. Communities meet others already under pressure. Some merge, some compete, some cease to leave descendants. Within one surviving network, an Rh negative related variant may happen to be carried by several families. It offers no visible advantage in the pursuit of deer. No protection clearly written on the landscape, no mark distinguishing its carriers as they sit around a hearth. Yet when their descendants multiply after the worst cold passes, the variant expands with them. Thousands of years later, researchers observe a higher frequency in a region and attempt to search for a dramatic cause. The dramatic cause may not be the trait itself. It may be the bottleneck and the survival of the people who happen to carry it. As the ice receded, Europe did not simply awaken into warmth. The transformation was unstable. Grasslands shifted. Forests spread across regions once dominated by open tundra steppe. Some large prey species declined or moved away. Coastlines changed as melting ice raised seas. Human communities followed new routes and adopted new strategies. Groups moved northward and eastward into territories opened by retreating glaciers, carrying with them the genetic legacy of southern refuges. But these movements were not one single way from one single shelter. Ancient DNA has revealed a far more complicated scene, different hunter-gatherer populations occupying parts of Europe at different times, replacing or mixing with one another, sometimes carrying ancestry that had survived in the west, sometimes connected to populations farther east. The continent was repopulated through multiple histories, not one clean migration. That detail matters whenever blood traits are linked too confidently to an Ice Age people. It is plausible that variants contributing to modern Rh negative frequencies circulated among prehistoric European groups. It is plausible that bottlenecks, refugees, and expansions helped shape their later distribution, but there is no preserved ledger identifying which Ice Age camp carried which Rh genotype in what proportion. Ancient DNA may eventually clarify more of the pattern. But Rh typing in prehistoric remains is not a simple matter of reading a modern hospital card from an ancient skeleton. Preservation varies. Samples are scarce and unevenly distributed. Population replacement and mixture can erase or reshape earlier signals, The evidence remains incomplete, and the map visible today cannot be projected backward without caution. After the Ice Age, another profound shift began to alter the genetic landscape. For thousands of years, Europe had been inhabited by groups whose lives depended primarily on hunting, gathering, fishing, and seasonal mobility. Then farming communities began moving gradually from Anatolia and the Aegean into southeastern Europe and onward across the continent. They brought cultivated cereals, domestic animals, new settlement patterns, pottery traditions, and demographic momentum. Farming could support large populations in many environments, though it also brought new disease burdens, nutritional challenges, and social pressures. The newcomers did not encounter an empty Europe. They met hunter-gatherer descendants whose ancestors had survived the Ice Age transformations. In some regions, the two populations coexisted for long periods. In others, mixture occurred earlier or more extensively, elsewhere. Farming ancestry became dominant while still absorbing local genetic contributions. A blood trait present among earlier hunter-gatherers might persist through that mixture, diminish, or become concentrated within communities that remained more isolated from expanding farmers. Conversely, variants brought by early farmers could enter Western populations and later appear ancient simply because they had been in Europe for several thousand years before written history. The genetic story does not respect the desire to assign every trait to one heroic source. The people of post-Ice Age Europe were already becoming mixtures of survivors and arrivals, each carrying older histories from landscapes beyond the continent itself. Several millennia later, the map changed again. During the late Neolithic and Bronze Age, ancestry connected with steppe pastoralist populations spread widely into Europe, particularly affecting northern, central, and western regions. These movements were associated with major social and cultural transformations, including new burial customs in many regions, changing mobility, and eventually the spread or consolidation of Indo-European languages across much of the continent. Although the relationship between genes, language, and culture is never perfectly one-to-one, in Iberia, steppe-related ancestry arrived and became especially prominent in paternal lineages, suggesting social processes that were not merely a gentle blending of equal populations. Yet even here, the incoming groups did not completely erase earlier inhabitants. They mixed with them, producing new communities from older and newer inheritances. By the time historical identities begin to emerge more clearly, the blood of Western Europe has already passed through repeated layers Ice Age survivors, post-glacial hunter-gatherer expansions, early farmers, Bronze Age migrants, and regional continuities shaped by geography and social practice. A higher frequency of Rh-negative blood in certain European regions could reflect any combination of these processes. It might preserve part of an ancient hunter-gatherer legacy. It might have been carried or altered through later populations. It might have risen locally through drift after the major mixtures were already complete. Most likely, no single movement explains everything. Human inheritance is rarely a solitary stream. It is a braided river separating, rejoining, absorbing tributaries, and leaving channels dry as landscapes shift. The same principle applies beyond Europe. Human groups across Western Asia, North Africa, Central Asia, and farther regions have always been connected through varying degrees of movement and separation. Climate opens corridors and closes them. Grasslands invite travel, deserts restrict it, coastlines offer routes, mountain passes filter who crosses. The genes associated with blood groups follow all of these paths. A variant can travel great distances through a small number of migrants, remain rare in most places, and become common only where demographic circumstances amplify it. Even a trait now most associated in popular imagination with Europe may have a history that cannot be reduced to European soil alone. The gene region itself is part of human biology with a far deeper evolutionary history, and the specific forms that create Rh-negative phenotypes have been shaped within diverse populations. There is a quieter human consequence hidden beneath all these movements. Each expansion remembered by genetics is also the loss or absorption of other lives. When new populations spread, earlier groups may survive through mixture rather than through the continuation of their languages or customs. A vanished people may not vanish biologically. Part of them may endure in descendants who no longer know their names, songs, roots, or rituals. A genetic variant can therefore outlast every cultural world that once carried it. The Rh-negative phenotype observed in a modern donor may have passed through populations whose tools changed, whose languages disappeared, whose sacred places were forgotten, and whose descendants later identified with entirely different communities. This is one reason rare bloodline stories feel so compelling. They begin with a real intuition. The body does preserve fragments of journeys no written history can recount. A blood test can touch something profoundly old. But the error comes in demanding that the fragment tell a simple tale of exceptional ancestry. Negative blood does not identify the carrier as a direct heir of one Ice Age people, nor does it separate that person from the wide network of human descent. It is instead one thread running through a woven fabric too complex to unpick by looking at a single strand. There may be ancient continuity within it, but also mixture, accident, loss, and survival shared by every human population. On a winter evening near the end of the Ice Age, a family huddles in shelter while wind moves over a landscape broader and colder than any living European city will ever know. A child sleeps wrapped in hide. Tools lie beside embers. Beyond the shelter, animals pass through darkness, following routes older than human memory. No one there knows about antigens, genes, donor registries, or antibodies crossing a placenta. No one knows that descendants, many thousands of years later. will study inherited differences and attempt to reconstruct the shape of the past from them. They know only that spring must come, that food must be found, that children must survive. Their bones, when they survive at all, will offer later scientists scattered clues. Teeth can preserve genetic fragments. Skeletons can hint at diet, injury, disease, migration, and burial practice. With new methods, even remains long silent can begin to reveal relationships between people separated by continents and millennia. If Rh negative blood has left a recoverable trail through prehistory, it is in these fragile remnants that the trail must be searched for, not in myths of superiority, nor in claims invented long after the people themselves vanished, but in the damaged, precious genomes of the dead. The dead do not offer their histories willingly. For most of the human past, they return to the earth almost completely, skin and blood gone first, possessions decayed, names unheard by any living ear. Sometimes a skeleton survives in a cave floor, a burial pit, a dry shelter, a lake margin, or beneath the foundations of a later village. Even then, the remains are not an open book. Bone is damaged by water, heat, soil chemistry, microbes, roots, excavation, storage. And the touch of modern people? DNA breaks apart after death into tiny fragments, and over thousands of years those fragments become scattered like ash. Yet within certain bones, particularly the dense inner portion of the bone near the ear, or inside teeth protected by enamel, traces of a vanished human life can persist long enough for science to retrieve them. This ability is recent enough that much of what earlier generations believed about prehistoric ancestry has had to be reconsidered almost in real time. For most of the 20th century, researchers approached ancient populations through tools, pottery, burial styles, skull measurements, language theories, and blood group frequencies measured in living people. These could reveal patterns, but they could not directly show whether people who made similar objects were closely related, whether a cultural change arrived with migrants or through adoption, or whether a modern population truly descended in isolation from those who occupied the same landscape thousands of years before. Ancient DNA altered that. A tooth from a grave can disclose that the individual buried there carried ancestry connected to farmers moving out of Anatolia. A bone from a Bronze Age cemetery can show the arrival of steppe-linked ancestry. A fragment from a hunter-gatherer can reveal that Europe once contained population patterns no longer present in the same form among the living. It is difficult to overstate the strangeness of this achievement. A person dies 5,000 years ago, their language vanishes, their village burns, erodes, or is buried beneath later soil. Their descendants intermingle with newcomers until no family can identify them by memory. Then, in a clean laboratory, a minute portion of bone powder is processed under strict precautions, because the DNA of the scientists themselves could easily overwhelm the ancient trace. Millions of short genetic fragments are sequenced and compared with those from other ancient individuals and living populations. Slowly, the dead begin to regain their place in the human map. They do not speak. They do not reveal beliefs or private grief, but they can show kinship, ancestry, biological sex, sometimes physical traits, pathogens, and patterns of movement invisible to archaeology alone. For the Rh negative mystery, ancient DNA seems at first like the instrument that should end all argument. If Rh status is genetic, and if ancient genomes can be read, why not simply test prehistoric individuals and discover where Rh negative blood began? The answer lies in the difference between detecting a variant and explaining its history. The genetics of the Rh system can be technically difficult. The Rhd and Rhce genes resemble one another closely, and their region of the genome is prone to deletions, rearrangements, hybrids, and variant forms. Ancient DNA is usually short, fragmented, and present in low quantities. Reading a complex, closely related gene region from damaged remains can be far harder than identifying broad ancestry patterns spread across thousands of genetic markers. Some ancient individuals can potentially be examined for variants associated with Rhd status, especially where genomic preservation and sequencing coverage are strong. But even a reliable result from one skeleton would say only that this one person likely possessed a certain genetic form. It would not identify the first appearance of the trait. It would not prove that a later population inherited its Rh negative frequency, primarily from that person's community. It would not show whether the trait was common, rare, or newly arrived in the larger population unless many appropriately sampled individuals existed from the same region and period. Prehistory rarely provides such generous evidence. Graves may be few, preservation uneven, and the individuals buried in particular ways may not represent the whole society. A map made from ancient remains is therefore never the past itself. It is a map of what has survived and what has been sampled. Cold, dry caves can preserve DNA better than humid soils. Elaborate burials may receive more archaeological attention than the resting places of ordinary people. Conflict, construction, erosion, looting, and changing sea levels erase entire communities before their genetic material can ever be recovered. The drowned coastal plains occupied after the Ice Age may hold crucial chapters of human movement beneath modern water. Regions rich in surviving samples can appear more important simply because they are easier to study. Regions with sparse preservation may seem silent not because nothing happened there, but because almost nothing remains available to read. What ancient DNA has revealed with far greater confidence is the larger population setting in which any Rh-related variants must have travelled. It has overturned the old image of prehistoric Europeans as one continuous people, slowly changing in place. Across millennia, hunter-gatherer groups with differing ancestry occupied the continent. Early farmers arrived and mixed with them. Later, steppe-related migrants contributed substantial ancestry to many regions. In Iberia, ancient genomes have helped show why the Basque case is not a surviving island from the deepest Paleolithic world, but a story of later continuity after major prehistoric mixtures. These findings matter for Rh-negative blood, even when no ancient sample neatly announces its Rh status, because they establish the roots, replacements, and mixtures through which any inherited blood trait would have had to pass. The surprising consequence is that living blood frequencies, once used as a major tool for reconstructing ancient populations, must now be interpreted in the light of ancient genomes, rather than used alone to define them. Earlier researchers could observe a high frequency of Rh-negative blood in a modern population and infer long isolation. Today, genetics can test whether that population truly remained isolated through the periods being proposed. Sometimes the broad intuition survives in modified form. A group may indeed show relative continuity during certain historical periods. Sometimes it fails. Populations assumed to be ancient holdovers turn out to contain major ancestry from later migrations. The laboratory has not removed mystery. It has made mystery accountable to evidence. This accountability becomes especially important wherever Rh-negative origin stories intersect with claims of hidden descent. If a modern group is said to preserve a unique ancient bloodline, then its genome should contain signals consistent with that history. If Rh-negative blood is claimed to come from a vanished population outside ordinary human ancestry, then ancient and modern genomic comparisons should eventually reveal an unusual contribution that cannot be explained by known human variation and migration. No credible evidence has shown such a separate origin. What ancient genomes reveal instead is the staggering interconnectedness of humanity lineages meeting after long separation, archaic human contributions embedded in modern genomes, populations expanding and contracting, and local identities forming from mixtures that later generations often imagined as pure. Ancient DNA also exposes the weakness of attaching fixed labels to people who never used them. A skeleton found in present-day France is not automatically French. A grave in modern Spain is not automatically Spanish or Basque. A prehistoric individual living near a later national boundary belonged to a world organised by kin, terrain, exchange, ritual, language, and movement in ways only imperfectly recoverable now. The same caution applies to blood. Finding a Rh-negative associated genotype in an ancient person from one region would not make that region the homeland of all Rh-negative people. Human variants can be older than the identities later associated with them, and can cross boundaries long before those boundaries exist. There is, however, a genuine allure in the possibility of finding the trait among the ancient dead. Suppose a well-preserved burial yields enough genetic material for researchers to infer that a person lacked the usual RHD gene. That individual, perhaps a woman who lived beside an Atlantic river 4,000 years ago, or a child buried with simple ornaments on a plateau, suddenly carries a feature recognisable in modern clinics. The discovery would not prove a conspiracy or confirm a superior lineage. Its power would be quieter. It would reveal a molecular connection between the living and a particular human being long removed from memory. The negative sign, printed beside a modern blood type, would be seen not as an internet mystery, but as something carried through actual lives, pregnancies, migrations, hunger, affection, illness, and burial. Even then, the most painful parts of the Rh story may remain almost impossible to detect. Could ancient DNA reveal a mother and fetus affected by Rh incompatibility? In exceptional circumstances, genetics might identify a potentially incompatible pairing, but proving that hemolytic disease caused a particular infant's death would be extraordinarily difficult. Many infants leave fragile remains. Disease may leave no distinctive mark on bone. The antibodies themselves do not wait in the grave for retrieval. A burial containing a woman and newborn can speak powerfully to human loss, but it cannot automatically reveal why the child died. The tragedy that modern medicine understands so well may have unfolded countless times in the past without leaving evidence precise enough to name it. This absence of proof is not absence of human reality. Prehistoric life included mothers who laboured, bled, survived, and mourned, infants who lived only hours or never breathed, families who buried the young with as much care as the old. Some of those deaths may have involved blood incompatibility. Most cannot be diagnosed. The limits of evidence require restraint, but they do not demand indifference. To read ancient genetics responsibly is to accept that the dead provide fragments, not complete confessions. The technology itself carries ethical weight. Human remains are not merely raw material for satisfying modern curiosity. They may be ancestors claimed by living communities, or bodies removed during periods of colonial collection and unequal power. Questions about who may authorise sampling, where remains should be held, how discoveries are communicated, and whether descendants should guide research are not secondary concerns. A genetic study can illuminate migrations while also reopening old wounds if it treats the dead as objects detached from living people. The search for ancient blood must therefore be guided not only by technical ability, but by respect. As methods improve, more precise answers may emerge. Researchers may recover Rh-related genotypes from wider ancient samples, compare frequencies across time, and test whether particular demographic events coincide with shifts in D-negative forms. They may discover that a pattern once attributed to one population movement arose through several. they may identify regional continuities now invisible among averages, or they may find that chance and later isolation explain more than any dramatic selective advantage. Scholars still debate how far future data will resolve these questions, because ancient sampling will always be imperfect, and human history will always contain losses no laboratory can reverse. For now, the bones do not point to a hidden race or a sealed origin. They point toward motion. They reveal farmers crossing into new lands, herders moving with livestock, hunter-gatherers persisting at the edges of expanding settlements, families blending ancestries across generations, and communities becoming distinct not because they stood outside humanity, but because they experienced its movements in particular ways. Somewhere along those paths, the absence of D travelled as well. Its route is not fully mapped. Its first carrier has no known grave, but its history, insofar as science can recover it, belongs among the ordinary dead who made the modern world by living through changes they could never name. And there is one more presence among those ancient genomes, one that has transformed the way people imagine human origins. Modern humans were not the only humans walking Ice Age landscapes. In bones older than farming, older than the cave paintings of Western Europe, older than nearly every familiar story of ancestry, another human lineage has yielded its DNA, the Neanderthals. Their genetic traces survive today in many living people. A fact so remarkable that it has drawn R-negative myths toward them almost inevitably. If modern blood carries an unusual absence, and if modern genomes carry fragments of extinct human relatives, perhaps the two are connected. It is a powerful suggestion, but power and evidence are not the same thing. The first Neanderthal whose bones entered modern science did not arrive with dignity. In 1856, workers quarrying limestone in the Neander Valley of Germany uncovered fragments of a skeleton from a cave, including a heavy-browed skull cap and thick limb bones. At first, even some educated observers resisted the idea that these remains belonged to a human form profoundly different from people living in Europe at the time. The bones were explained away as those of a deformed modern person. Perhaps a diseased individual, perhaps a soldier from some earlier conflict. The deeper implication was uncomfortable. If the skeleton belonged to another kind of human, one that had inhabited Europe long before recorded history, then the human story was not a single uninterrupted line leading neatly to the present. It was a landscape once shared with close relatives who looked different, lived differently in some respects, and eventually disappeared. More than a century and a half later, the Neanderthals have moved from caricature to complexity. They were not the shambling dim creatures once imagined in popular pictures. They made sophisticated stone tools, controlled fire, survived cold and unpredictable environments, hunted large animals at close range, used pigments and ornaments in some contexts, cared for individuals who lived with serious injuries, and buried at least some of their dead. Though the extent and meaning of such practices remain debated, their bodies were adapted to a demanding Eurasian world, powerful, compact, broad through the chest and pelvis, with faces and skulls shaped differently from those of modern humans. For hundreds of thousands of years, Neanderthal populations occupied parts of Europe and Western Asia, while the ancestors of modern humans developed largely in Africa. Then the paths of these human lineages met. The encounters did not end only in competition or disappearance. They also produced children. Genetic evidence has made that fact unavoidable. Many living people with ancestry outside sub-Saharan Africa carry a small percentage of Neanderthal-derived DNA inherited from interbreeding that occurred after modern human populations expanded into Eurasia. The exact proportion varies among individuals and populations, and ancient events were likely more complicated than one isolated meeting. There may have been several episodes of contact, followed by natural selection removing some Neanderthal-derived DNA, while preserving other fragments. The extinct are not entirely gone. Parts of them remain dispersed across living genomes, not as a complete Neanderthal identity in any one person, but as inherited fragments carried by millions of people. It is one of the most extraordinary discoveries in the study of human origins, and it almost immediately became attached to Rh-negative blood. The association seems, at first, almost irresistible. For negative blood is rare, invisible, and often described as ancient. Neanderthals are extinct, enigmatic, and genetically present within modern humans. Join the two ideas together, and a dramatic story forms itself. Perhaps Rh, negative people descend especially strongly from Neanderthals. Perhaps the missing D antigen is a surviving marker of interbreeding. Perhaps a trait once carried by another human lineage now identifies their closest living heirs. The appeal of that story is real, the evidence for it is not. One of the simplest problems is that Neanderthal ancestry is not confined to Rh-negative people. Neanderthal-derived DNA is found broadly among populations in Eurasia, and among many populations elsewhere, whose ancestry includes later Eurasian gene flow. Most of these people are Rh-positive, in large parts of East Asia, where Neanderthal ancestry is present, and in some comparisons appears at least as high or slightly higher than in Europeans. Ordinary Rh-negative blood is extremely rare. If Rh-negative status were a direct and distinctive marker of Neanderthal inheritance, those broad population patterns would be difficult to reconcile. The map of Neanderthal ancestry and the map of Rh-D negativity are not the same map. There is another problem. Neanderthals did not carry a single known blood identity that can simply be placed against a modern hospital label. Ancient genomes have allowed researchers to investigate blood group-related genes in some archaic humans, and those results suggest that Neanderthals possessed their own variation within systems including ABO and Rh. They were not a uniform population of one mysterious blood type. Nor were they biologically removed from the basic architecture shared by modern humans. The Rh system existed within the wider evolutionary history of hominins. Its variance cannot be reduced to the idea that one antigen absent suddenly appeared only because two wholly separate kinds are being mixed. Some archaic Rh-related genetic findings are intriguing, and scholars continue to refine their interpretation as additional high-quality ancient genomes become available. But nothing establishes ordinary modern Rh-negative blood as a uniquely Neanderthal inheritance. Even the word Neanderthal can mislead when used as though it refers to a single, unchanging population. Neanderthals lived across enormous stretches of time and space, from Western Europe to parts of Central Asia. Communities separated by thousands of years and wide distances were not genetically identical. Their populations were often small, and some show signs of inbreeding or limited diversity. A blood-related variant present in one individual or group would not automatically characterize all Neanderthals. Any more than a variant observed in one ancient modern human would define every human alive during the same age. The dead rarely offer the clean categories the imagination wants. Modern humans, too, were more complicated than the older story of a triumphant replacement once suggested. When groups of Homo sapiens entered landscapes inhabited by Neanderthals, they did not simply encounter an alien species and sweep it away untouched. They exchanged genes, perhaps exchanged knowledge in ways impossible to reconstruct, and lived through overlapping ecological pressures. Some Neanderthal-derived genetic variants in living humans have been associated with aspects of immunity. Skin biology, metabolism, pain sensitivity, sleeping patterns, and responses to disease. Some may once have helped arriving modern humans adjust to Eurasian environments already familiar to Neanderthals. Others may have carried costs or become disadvantageous in later settings. Many fragments are simply small surviving remnants whose effects, if any, are difficult to interpret. This is the real significance of archaic inheritance, not that it produces a special class of modern person, but that it destroys the illusion of a perfectly separate and linear human past. A person may carry Neanderthal-derived fragments without looking unusual, feeling unusual, or belonging to any mystical inheritance. Those fragments are spread among ordinary people who speak different languages, live on different continents, and possess every variety of ABO and Rh blood type. Archaic ancestry is not a badge visible only in rare blood. It is part of the shared, unevenly distributed complexity of modern humanity. The claim that Rh-negative people are especially Neanderthal can also drift into a second, contradictory legend. In one version, Neanderthals are treated as primitive, and therefore Rh-negative people are imagined as strange relics from an older form of humanity. In another version, Neanderthals are reinvented as unusually perceptive, spiritually powerful, physically superior ancestors whose supposed qualities survive in rare blood descendants. Both versions place modern fantasies on ancient people. Neanderthals were neither brutish failures nor secret superhumans. They were human relatives extraordinarily adapted to their own worlds, capable of suffering, skill, endurance, and social life, whose disappearance after modern humans spread through Eurasia remains one of prehistory's most profound losses. Why, then, did they vanish? Climate instability may have placed severe pressure on already small and scattered populations. Competition with expanding Homo sapiens may have mattered, including differences in social networks, technology, resource use, or population size. Interbreeding may have absorbed parts of Neanderthal ancestry into larger modern human populations, even as distinct Neanderthal communities disappeared. Disease transmission may also have played a role. No single explanation fully accounts for their extinction across every region, and the ending may not have been one sudden event, but a long, uneven fading, with local populations declining at different times and under different pressures. Their genes survived partly because their disappearance was not complete separation followed by total erasure. Some of their descendants were also descendants of modern humans, folded into a population that continued onward. There is a haunting quality to this kind of survival. A people can disappear as a recognizable population, while fragments of their bodies continue in people who never know their names except through science. A gene involved in immune response, a small contribution to skin or metabolism, a stretch of DNA carried without awareness, these are not memories in the human sense, but they are survivals. They show that extinction among closely related humans can be less like a door closing completely, and more like a voice disappearing, while a few syllables remain scattered through later languages. Our negative blood invites a similar emotional response because it, too, is silent inheritance. It is natural that the two should be joined in popular imagination. Both appear to connect modern bodies with deep time. Both carry the suggestion that something unseen has survived, yet the responsible path is to keep the stories distinct unless evidence joins them. Neanderthal ancestry is real, or negative blood is real. Their existence in the same broad human history does not mean one explains the other. A modern Rh negative person may carry Neanderthal-derived DNA, just as a modern Rh positive person may. The antigen missing from the red blood cell does not provide a shortcut to identifying archaic ancestry. There may still be scientific questions at the edges. The evolution of Rh-related genes among archaic and modern human populations is a legitimate field of inquiry. Researchers can compare ancient gene variants, study whether unusual blood group patterns influence pregnancy or survival in small prehistoric populations, and examine how blood systems changed as human lineages encountered one another. A particularly intriguing possibility is that reproductive incompatibilities involving blood group variants could have created difficulties in some archaic modern human pregnancies, though any broad historical role remains uncertain and must not be overstated. Ancient genomes may eventually clarify more. At present, they do not support the claim that ordinary Rh negative status is proof of direct Neanderthal descent or a marker of a concealed archaic bloodline. The difference between inquiry and myth lies in the willingness to accept an answer that is less sensational than the question. The Neanderthals add immense depth to the Rh negative story not because they reveal the secret origin of the negative sign, but because they remind us how many vanished populations contribute to the world of the living in ways that are partial, irregular, and difficult to trace. Our bodies are not sealed records with one secret label stamped inside. They are collages made by survival modern human lineages shaped in Africa, migrations into new continents, meetings with archaic relatives, local bottlenecks, expansions, isolation, famine, fertility, disease, and millions of ordinary births. In a cave somewhere in Ice Age Eurasia, a Neanderthal family once gathered around a fire. Their blood circulated through bodies adapted to cold, carrying antigens and genes whose precise combinations may never be known. Elsewhere, perhaps in another valley or beyond a mountain range, early modern humans carried their own variations. At some point, their histories touched closely enough for ancestry to cross between them. No one standing there could have imagined blood banks, genetic sequencing, or the future hunger to classify descendants by what survived inside their cells. They lived in the present tension of cold, food, kin, injury, and continuation. Tens of thousands of years later, the Neanderthals have been recruited into stories they never authored, stories of hidden powers, forbidden descent, rare blood, and human beings supposedly divided into secret kinds. But their true legacy resists those divisions. Their DNA demonstrates contact, mixture, and shared vulnerability. It shows that even the line once drawn between different human forms could be crossed, and if this does not explain the Rh negative trait, it places that trait inside a much larger truth: the past within human blood is not a certificate of superiority. It is evidence that survival has always depended on entanglement. Yet myths rarely retreat simply because a scientific connection fails. When a rare trait cannot be securely tied to Ice Age survivors or Neanderthal inheritance, the appetite for meaning often searches elsewhere. The negative sign begins to gather qualities no test has ever found: unusual intelligence, heightened intuition, resistance to ordinary influence, strange physical characteristics, an instinctive sense of not belonging. The blood leaves the laboratory altogether and enters a new domain, one in which rarity is no longer merely inherited difference, but identity, destiny, and proof that the carrier is set apart. Once a difference has been made visible, people rarely leave it inside the laboratory. A blood test may begin as a clinical fact, a notation in a chart, an instruction to a hospital, a precaution in pregnancy. But to the person who receives the result, especially when the result is described as uncommon, it can begin to feel like an answer waiting for a question. For a negative, a small absence on red blood cells, a trait carried from parents, perhaps shared by some relatives and not others. A label connected with medical danger, unusual distribution, and stories of ancient survival. In an age flooded with information and uncertainty, that combination is almost designed to acquire a mythology. The mystery no longer needs a cave, a mountain refuge, or a prehistoric grave. It begins in the private moment when someone types a blood type into a search bar and discovers not only medicine, but an identity offered back to them. The claims tend to appear in clusters. Rh negative people are said to have unusually high intelligence, sharper intuition, heightened sensitivity, exceptional empathy, vivid dreams, psychic awareness, low body temperature, unusually coloured eyes, additional ribs or vertebrae, resistance to conventional authority, or a persistent feeling of being different from those around them. Some lists describe them as natural leaders, others as loners. Some portray them as spiritually advanced, others as biologically anomalous. Nearly all share the same pattern. The trait is presented not simply as a blood group variation, but as a secret explanation for a person's temperament, body, life struggles, and private sense that they do not quite belong. There is no credible scientific evidence that ordinary Rh negative status creates these psychological powers or physical attributes. A missing D antigen does not establish superior intelligence, extrasensory perception, unusual emotional depth, or a fixed personality type. Eye colour, body temperature, bone structure, and neurological traits are shaped through many genes: development, environment, health, and ordinary human variation. They are not bundled into a hidden syndrome by the absence of RhD. Rh negative people can be perceptive or inattentive, calm or anxious, scholarly or practical, warm or distant, healthy or ill, just as Rh positive people can. The blood trait matters strongly in certain medical circumstances, but it is not a biological script for an entire person. That statement is simple. The reasons people reject it are not. Rarity has a psychological gravity of its own. Most people go through life as part of categories they did not choose: a generation, a hometown, a profession, a language, a family history, a social class, a body with its particular strengths and vulnerabilities. An unexpected rare trait offers a different kind of belonging. It suggests that something inside the person has always been exceptional, whether or not the wider world recognized it. If someone has felt isolated, intensely sensitive, misunderstood, restless, or drawn to mysteries. A list of supposed Rh negative characteristics can feel less like random internet content and more like recognition. The list does not need to be accurate in a scientific sense to feel personally exact. It is only to include enough broadly human experiences that a reader can see a reflection there. This is the mechanism behind many personality myths. Traits such as vivid dreaming, feeling different, questioning authority, sensing danger, having moments of strong intuition, or being unusually affected by the moods of others are not rare in human life. They are especially powerful when described in flattering or mysterious terms. People remember matches and disregard mismatches. A reader who identifies with 6 qualities on a list may not notice that the list is broad enough to include an enormous portion of the population. If the identity is appealing, the mind naturally begins gathering supporting evidence. A family member who is also independent becomes confirmation. A strange coincidence becomes a sign. An ordinary medical detail becomes an origin story. The Rh negative myth gains further strength from the fact that the trait is not invented. Unlike a horoscope category or a fictional order of initiates, Rh status can be tested in a laboratory. It is recorded by hospitals. It affects real pregnancies and transfusions. It differs in frequency across populations. All of that gives the surrounding unsupported claims a borrowed authority. When a story begins with true medical facts, its later leaps may feel more credible than they deserve. The line between evidence and imagination becomes blurred not because the underlying science is false, but because it is used as a foundation for claims it never actually supports. A person may learn that Rh negative mothers once faced serious pregnancy risks, then encounter a site suggesting that the trait is incompatible with ordinary humanity. They may learn that frequencies are elevated in certain populations, then encounter a claim that the carriers descend from an advanced lost civilization. They may read that the Rh system is biologically complex, then be told that scientists are unable to explain it because its true origin is being concealed. Each move takes a real observation and changes its meaning. Medical consequence becomes proof of alienness. Regional frequency becomes proof of ancient rank. Scientific uncertainty becomes evidence of suppression. The result feels like investigation, but it is built by turning gaps in knowledge into conclusions. For those who carry the trait, this can produce a curious emotional double movement. On one side is anxiety. Rh negative blood becomes something dangerous, unnatural, potentially incompatible with pregnancy, or somehow marking the person as biologically separate. On the other side is elevation. The person is offered special abilities, secret ancestry, and a role within a hidden minority that knows what others do not. Fear and pride reinforce one another. To be told one's blood is unusual can be unsettling. To be told its unusualness signifies hidden importance can make that unease rewarding. Mystery supplies dignity to uncertainty. Long before the internet, human societies attached meaning to blood. Blood was kinship, oath, purity, pollution, inheritance, sacrifice, violence, kingship, curse, and redemption. Families imagined nobility in blood. Communities imagined outsiders as carrying blood of another kind. Rulers claimed descent through sacred or superior lines. None of these beliefs required a microscope. The modern blood group offered old symbolism and new scientific costume. Now difference could be printed on a card. It could be named with letters and signs. It could be measured statistically. What had once been metaphor could be made to seem biological, and what was genuinely biological could be made to carry moral or spiritual weight. The history of such thinking demands caution. Claims about inherited superiority have rarely remained harmless curiosities. When societies begin treating blood as evidence of deeper human worth, some people are inevitably raised while others are diminished. The Rh negative mythology often presents itself as a private puzzle rather than a political doctrine, but phrases such as superior bloodline, pure ancestry, chosen descendants, or non-ordinary humans echo older and darker ideas. They reduce the complexity of persons to inherited markers and treat accident of birth as destiny. Even when intended as praise, biological exceptionalism is a narrow room in which to place a human life. There is another subtle arm. A mythology of extraordinary difference can distract from the real importance of the blood type. Rh negative donors are valuable, not because they possess hidden abilities, but because, in particular clinical circumstances, the red cells may be urgently needed. Pregnant patients require accurate care not because their bodies are mysterious or abnormal, but because preventable immune sensitization can threaten a fetus. A person who believes online claims may become afraid of standard medical information, distrust screening, or misunderstand why preventive treatment matters. The imagined secret begins to crowd out the practical truth that saves lives. The internet does not create this pattern from nothing, but it accelerates it. A claim once passed quietly between a few enthusiasts can now be repeated through videos, forums, social feeds, automated recommendations, and copied articles that cite one another rather than primary evidence. A person searches for the meaning of a blood test and is soon offered a cascade of increasingly dramatic explanations. Platforms reward curiosity, surprise, and emotional reaction. A careful account of red cell antigens may be medically valuable, but a title promising hidden origins or forbidden traits provokes more clicks. Repetition then begins to resemble confirmation. When the same idea appears on dozens of pages, it can feel as though many independent sources have verified it, even if all are recycling the same unsupported claim. Some storytellers go further and collect ordinary observations into a supposed profile. A carrier may report a low resting temperature, and it becomes characteristic of the blood type. Someone with Rh-negative blood describes a powerful intuition, and it is added to the list. A blue-eyed person joins a discussion. An eye colour is woven into the pattern. Missing are the Rh-negative people who do not match, the Rh-positive people who do, and the controlled comparisons needed to establish whether any association is real. Anecdotes are not worthless as human testimony, but they do not become genetics merely by accumulating online. Without rigorous evidence, a list of traits is a portrait assembled after the subject has already been declared unusual. Science is sometimes blamed for sounding unsatisfying in response to someone asking why they have always felt different. The answer that Rh-negative blood is mainly relevant to transfusion and pregnancy may seem emotionally inadequate. It explains the antigen but not the life. It leaves untouched the loneliness, creativity, unusual experiences, family estrangements, or intense intuitions that made the person seek a larger account. But the failure of a blood type to explain those things does not make them unreal. Human experience can be profound without being encoded in RhD status. A person may be extraordinary through character, history, insight, suffering, work, or imagination. None of that needs permission from a red cell antigen. There may be legitimate questions about subtle medical associations of Rh status. Researchers have at times investigated whether Rh phenotypes interact with particular infections, neurological responses, or other health outcomes. Such studies can be difficult to interpret, may produce mixed results, and do not support sweeping descriptions of a rare blood personality or hidden power. An association explored in a research context is not a license to declare that millions of people possess a common supernatural profile. Scholars still debate some possible biological relationships beyond the well-established transfusion and pregnancy consequences, but the evidence does not turn Rh-negative individuals into a distinct class of mind or spirit. The strongest mystery remains the transformation itself, how readily a surface antigen becomes a story about the self. A person begins with blood, moves to ancestry, then to meaning. The path feels intimate because blood is inside the body, inherited and involuntary. It seems more truthful than fashion, more permanent than belief, more private than a name. If some ancient survival can be found in it, then perhaps one's own unexplained feelings appear anchored to history. Yet blood cannot carry every answer asked of it. The negative sign is old, medically significant, and unevenly distributed. It is not a confession from the universe about who deserves to feel special. Still, ordinary rarity was never likely to be the end of the story. Once Rh-negative blood had been made to signify difference, another step followed naturally. If carriers seemed unusual, perhaps their ancestors had not been ordinary humans at all. The theories began reaching beyond Ice Age survivors and Neanderthals toward beings of legend and authority, fallen angels, gods descending from the sky, royal dynasties guarding a separate inheritance, visitors from other worlds whose descendants supposedly remain among us. In these stories, blood is no longer only an identity. It is evidence of an intervention hidden at the beginning of civilization, and of a secret protected ever since. Once blood is imagined as evidence of hidden identity, it does not take long for the story to leave humanity altogether. The missing antigen becomes a doorway. Behind it gather beings descending from the sky, angels expelled from heaven, gods breeding with mortals, serpent dynasties, sacred kings, secret aristocracies, and visitors from distant worlds whose descendants are said to remain quietly scattered among ordinary people. In these stories, Rh-negative blood is not merely rare and medically important. It is presented as a biological signature left behind by an ancient intervention, a sign that some modern humans carry an inheritance fundamentally different from everyone around them. The idea is dramatic because it offers three things at once: a mystery of origin, a hidden hierarchy, and the thrill of believing that official history has overlooked or actively suppressed the most important clue of all. Ancient texts are frequently drawn into this web. The most common references come from traditions describing divine beings interacting with human life, the Nephilim in the Hebrew Bible, the sons of God and daughters of men, the demigods of Greek myth, divine kings in Mesopotamian traditions, extraordinary heroes born from unions between gods and mortals, and later legends of royal families claiming sacred ancestry. And these stories are real as cultural artifacts. Ancient societies did imagine the boundary between gods and humans as permeable. Kings linked themselves to divinity because authority is stronger when it appears written into the order of the universe. Heroes were given supernatural parentage because their achievements seemed too large for ordinary origins. But these traditions are not genetic records. They do not describe the Rh blood group, the D antigen, chromosome 1 maternal sensitization, or any biological mechanism that can connect them to modern Rh-negative people. The leap is usually made by resemblance rather than evidence. A myth mentions beings from above, so those beings are reinterpreted as extraterrestrials. A ruler claims descent from a god, so that claim is reinterpreted as a literal breeding program. A modern blood trait is rare and inherited, so it is assigned to the supposed offspring. The argument then circles back on itself. The ancient stories prove unusual, ancestors existed, and the unusual blood proves the stories were factual. But neither part actually supports the other. Myths can preserve memories of events, environments, rulers, disasters, and social fears, sometimes in altered form, but they cannot be treated as laboratory reports merely because a modern theory finds their imagery convenient. The figure of the Nephilim has become particularly useful in these narratives. In a brief and enigmatic biblical passage, they appear in a world moving toward catastrophe, associated with figures of ancient renown. Later interpretive traditions expanded their meaning considerably. Some saw them as giants, some connected them with fallen angels, some placed them within elaborate cosmologies of corruption and judgment. In modern speculation, they are sometimes recast as non-human visitors whose descendants supposedly carry Rh negative blood, but the scriptural text does not mention blood groups, nor does any credible historical or genetic evidence bridge that gap. A tradition shaped by religious meaning, ancient storytelling, and later interpretation is being asked to function as a coded description of immunohematology. The demand tells us more about modern hunger for hidden ancestry than it does about ancient blood. Mesopotamia offers another rich source for reimagining. The civilizations of Sumer, Akkad, Babylon, and Assyria left behind myths in which gods create, punish, reward, fight, appoint kings, and structure the cosmos. Because these societies were ancient, technically impressive, and once written in scripts most modern people cannot read, they are especially vulnerable to being recruited into theories of secret knowledge. The gods are transformed into alien engineers. Creation narratives become genetic experiments. Kingship becomes proof of controlled hybrid descent. Yet specialists who read the actual surviving tablets work with languages, genres, scribal traditions, political theology, and damaged fragments whose meanings require careful reconstruction. Those texts are extraordinary precisely as human works attempts to understand order, mortality, flood, labour, power, and the relationship between a vulnerable city and the divine world imagined above it. There is no credible tablet linking the RHD antigen to an ancient non-human lineage. The same pattern follows royal blood. Across history, rulers have claimed exceptional descent. Egyptian pharaohs were embedded in divine kingship. Roman emperors could be associated with divine ancestry, and later deified. Medieval European dynasties constructed pedigrees connecting themselves with legendary founders, biblical figures, Trojans, saints, or heroes. Royal marriage often remained within restricted social circles, sometimes producing very visible inherited disorders. This makes royal bloodlines an attractive setting for theories of preserved Rh-negative ancestry. If the trait is uncommon, and elites married elites, perhaps they deliberately protected a secret inheritance. Perhaps their claims of divine right were not merely political language, but faint memories of biological difference. There are several problems with this claim. First, no secret breeding scheme is needed for a hereditary trait to appear within aristocratic families. Any inherited blood type can circulate through lineages, and marriages among related or socially limited groups can increase the visibility of many traits. Second, rulers and nobles have not been shown to carry a single exclusive Rh-negative lineage, distinguishing them from wider populations. Third, royal claims to sacred descent served obvious political and cultural purposes. A king who declares divine ancestry is strengthening legitimacy, not offering a coded blood bank document to future investigators. Finally, the actual genetic consequences of elite intermarriage, where well documented, are often far less glamorous than myths suggest fertility problems, inherited disease, frailty, and the magnification of harmful variants rather than the preservation of hidden perfection. Perhaps the most adaptable modern theory is extraterrestrial origin. It has the advantage of being able to absorb almost any evidence. If Rh-negative blood differs from the majority, that difference is presented as proof of alien contribution. If scientists explain the genetics, the explanation is dismissed as a cover story. If no unusual physical traits are found, the inheritance is said to be subtle. If ancient DNA fails to reveal a separate origin, the intervention is placed in a period or form not yet detectable. A theory that treats every contradiction as evidence of concealment becomes nearly impossible to disprove, but only because it has stopped behaving like an evidence-based claim. It survives not by predicting discoveries that can be tested, but by retreating whenever testing fails to support it. A genuine non-human genetic contribution to modern humans would not need to be inferred from a single common blood group phenotype. It would be a profound genomic event. Human genomes contain evidence of ancestry, population separation, interbreeding, and shared descent. The discovery of Neanderthal and Denisovan ancestry became possible because ancient and modern DNA carried detectable patterns that could be compared, tested, replicated, and debated openly. An unknown non-human lineage contributing uniquely to Rh-negative people would be expected to leave broader genetic signatures, not merely the absence of a red cell antigen that can be explained through known human genetic variation. No credible genomic evidence has revealed such a contribution. The Rh-negative phenotype fits inside the human range, complicated though that range is. The absence of such evidence does not mean science has proven that intelligent life exists nowhere else in the universe, or that every ancient mystery has been fully solved. The question of life beyond Earth is a legitimate scientific question, approached through astronomy, planetary science, chemistry, and the search for detectable biosignatures or technological signals. But the possibility of life elsewhere cannot be used as evidence that a particular human blood type arose from alien ancestry. Possibility is not proof. A universe vast enough to inspire wonder is not a blank check for attaching extraterrestrials to every rare feature in human biology. The angelic and alien accounts share an emotional structure. They begin by separating carriers from ordinary humanity, then elevate that separation into purpose. Or negative people are not only different, they are descendants of watchers, gods, star beings, or ancient rulers. Their medical trait becomes a secret nobility. This can be alluring for anyone who has felt alienated, overlooked, or drawn to the idea that their life contains a concealed significance. Yet it carries the same fundamental danger as every claim of special blood it mistakes an inherited feature for a measure of human value. Whether the imagined ancestors are royal, divine, or extraterrestrial, the result is a hierarchy written into bodies. History offers enough warning about what happens when groups are described as carrying superior or polluted blood. Not every online discussion of Rh negative origins is malicious. Many people are simply curious, searching for meaning in a genuinely unusual medical fact, but the language of bloodline secrecy easily drifts toward exclusion. It can become entangled with ideas about chosen races, hidden rulers, contaminated populations, or elites who control society through inherited biology. Some theories reverse direction and portray Rh-negative people not as superior but as dangerous outsiders, non-human infiltrators, or evidence of sinister manipulation. Admiration and suspicion are closer relatives than they appear. Both begin by denying that a person with a rare trait is simply human. The archaeological record tells a different story. Ancient people were capable of astonishing engineering, mathematics, observation, navigation, art, ritual, and political organization without requiring instruction from visitors from the stars. Pyramids, megaliths, irrigation systems, cities, calendars, astronomical alignments, metalwork, ships, and written epics emerge from human minds working across generations within particular environments and societies. Explaining these achievements through alien intervention does not honor ancient people, it diminishes them. In much the same way, explaining Rh-negative blood through supernatural descent strips away the true scale of human inheritance. Ordinary humans, surviving ordinary and extraordinary pressures across immense periods of time, are capable of producing remarkable genetic patterns without assistance from beings outside the species. This does not mean ancient beliefs should be mocked or discarded. The myths recruited into Rh-negative theories deserve attention in their own worlds. A flood narrative may reveal how river civilizations experienced catastrophe and moral order. A divine birth story may reveal how societies understood kingship, heroism, or legitimacy. An account of beings crossing boundaries between heaven and earth may embody anxieties about power, desire, violence, and the fragile distinction between the human and the sacred. These stories have depth. Reducing them to scrambled references to blood antigens or extraterrestrial breeding removes much of what made them meaningful to the people who told them. There is also something telling in the fact that Rh-negative myths so often seek ancestors above humanity rather than within it. The real history of blood is crowded with anonymous people, mothers who lost infants, donors whose blood rescued strangers, isolated communities preserving variants by chance, migrants crossing difficult terrain, patients waiting for compatible units, scientists examining cells under controlled light. Their lives may not promise cosmic privilege, but they are not less profound. A molecular absence carried through thousands of years by ordinary people is not a weak story. It is only less flattering to the desire for exclusivity. What remains uncertain is not whether aliens, angels, or divine dynasties produced Rh-negative blood. There is no credible evidence that they did. The honest uncertainties are more grounded and, in their own way, more demanding when particular Rh-negative genetic forms first arose, how their frequencies changed among ancient populations, what balance of drift, founder effects, mixture, and possible selection shaped their distribution. How many personal tragedies before modern medicine were caused by incompatibility that no one could recognize? These are questions that can be investigated, even if answers remain partial. They ask for evidence rather than allegiance. Still, unsupported explanations thrive where distrust has already prepared the ground. A person may accept that myths alone do not prove an alien bloodline, yet continue wondering why the truth seems hard to find. Why is the subject wrapped in technical language? Why do hospitals collect and track rare blood? Why do some facts seem absent from ordinary education? Why did medicine take so long to prevent Rh-related infant death? Once a story of secrecy begins, every institution surrounding blood can be made to look suspicious. The next turn in the Enigma does not depend on ancient gods or visitors from another world. It depends on something far more familiar, the uneasy relationship between ordinary people and the systems that ask to be trusted with their bodies. Trust is easy to demand from a patient and difficult to rebuild once it has been broken. Blood makes that problem sharper than almost anything else in medicine. It is removed from the body, measured, labelled, stored, transported, transfused, tested, and discussed in a language most people never learn unless a crisis forces them to. A donor gives blood and rarely knows who receives it. A pregnant woman is told that an injection may protect a future child from antibodies she cannot feel and has never seen. A patient in an emergency is given units selected by unseen technicians working behind locked laboratory doors. For these systems to function, ordinary people must accept that strangers understand something intimate inside them and will handle it correctly. Most of the time, that trust is rewarded with care so effective that the danger disappears from view. But when people already suspect that important truths are being hidden, the very efficiency of the system can begin to look like evidence of control. A blood bank is a practical institution, yet to an outsider it can seem filled with the ingredients of secrecy. Donor types are recorded. Rare donors may be contacted repeatedly. Some units are reserved for particular patients or frozen for unusual compatibility needs. Hospitals track antibodies, pregnancies, transfusion histories, and cross-matching results. Certain blood is described as especially valuable. None of this requires a hidden agenda. A patient who has formed antibodies after pregnancy or previous transfusions may be unable to receive ordinary matched blood safely. A person with rare blood may be one of very few compatible donors for another patient. A hospital that fails to maintain such records is not protecting freedom. It is risking death. Yet language such as rare, registry, compatible, reserve, and special donor can easily be rearranged into a darker narrative by anyone already primed to believe that blood has concealed importance. The technical nature of the subject deepens the distance. The ordinary patient hears terms such as antigen, antibody screen, sensitization, hemolysis, genotype, phenotype, weak D, partial D, prophylaxis, and alloimmunization. Each word is precise within medicine, but precision does not always feel reassuring when it is delivered quickly in an anxious room. A pregnant woman may hear that she is Rh negative and needs anti-D immunoglobulin because her baby might be Rh positive. She may understand the immediate instruction but still leave with a disturbing impression her blood is somehow at odds with her child. If she later encounters an online account claiming that Rh negative mothers are evidence of a separate human lineage, the full story may seem to explain the emotional discomfort left behind by the clinical encounter. Medicine may have provided correct care without providing a narrative a frightened person can comfortably carry. This is one reason conspiracy stories flourish around real complexity. They offer simpler causes and more dramatic meanings. Instead of many genes, there is one secret bloodline. Instead of carefully stored donor units, there is a hidden interest in rare blood. Instead of an immune system reacting to an antigen, there is proof of incompatibility between different kinds of humans. Instead of scientific uncertainty about ancient frequency patterns, there is deliberate silence. The conspiracy version is wrong, but it is often psychologically tidy. It removes confusion by replacing it with intention. Someone knows, someone hides, someone benefits. Random inheritance, demographic chance. A molecular accident may describe reality more accurately, but they do not satisfy the part of the mind that feels safer when events have an author. It would be comforting to say that distrust of medical systems is born only from misunderstanding. History does not allow that comfort. Medical institutions have at times betrayed the very people they claim to serve. Research has been conducted without proper consent. Samples have been taken and used without patients being informed or allowed control over what came from their bodies. Marginalized people have been deceived, neglected, exploited, or treated as material for knowledge rather than as human beings with rights. In the United States, the Tuskegee syphilis study allowed black men with syphilis to go without proper treatment, while researchers observed the course of disease, even after effective therapy existed. The cells taken from Henrietta Lacks became one of the most important tools in modern biomedical research, while she and her family were long excluded from meaningful knowledge or consent concerning their use. These histories are not rumours, they are documented failures of ethics and justice. Blood services, too, carry painful histories. During the 20th century, contaminated blood products infected patients in multiple countries, with viruses including HIV and hepatitis C. Many of the affected patients required blood products for conditions such as haemophilia or received transfusions during medical care. They entered the system seeking treatment and emerged with life-changing or fatal infections. Investigations and public reckonings have shown how delayed action, poor oversight, institutional defensiveness, financial pressures, and failures of transparency could deepen the harm. For families who watched loved ones become ill after receiving something meant to preserve life, suspicion was not irrational paranoia. It was grief sharpened by the knowledge that institutions had failed them. Such events do not prove that Rh negative blood conceals an alien origin, a royal breeding program, or suppressed genetic powers. But they explain why reassurance alone sometimes fails. A person who knows that medical authorities have lied before may not be calmed by being told simply to trust authorities now. A person whose community has experienced unethical treatment may reasonably demand evidence, clarity, accountability, and consent. The answer to false claims is not to pretend institutions have always behaved perfectly. It is to separate documented wrongdoing from unsupported speculation. There have been real cover-ups in medicine. That fact makes it more important, not less, to distinguish them from theories that attach secrecy to Rh negative blood without credible evidence. There is a significant difference between a secret and a specialty. Much of transfusion medicine feels obscure because it is specialized. Blood group systems beyond ABO and RHD are rarely discussed in ordinary schooling because most people will never need to interpret them personally. Hospitals do not display every compatibility protocol in public waiting rooms because the procedures are carried out by trained laboratories under regulated conditions. Just as pharmacies do not teach every patient the full chemistry of every medication before dispensing it. This distance can appear ominous when viewed through the lens of suspicion, but it is not in itself concealment. The Rh system is taught in medical education, discussed in scientific literature, explained in patient information, investigated in genetics, and managed openly through clinical guidelines. Its complexity is accessible, though not always easily digested. The tracking of rare blood also has an entirely human reason. Imagine a patient whose immune system has formed several antibodies, making most donor blood unsafe. A routine transfusion becomes a search for the few units that will not trigger destruction of red cells, but somewhere, perhaps in another city or another country, a donor with compatible blood is given a unit that can be stored, transported, and delivered when required. A registry allows that match to occur. The system does not value the donor as a superior person or seek to harvest an extraordinary substance. It values a compatibility pattern that might save a patient with equally uncommon needs. In that exchange, rarity produces responsibility rather than rank. For Rh-negative donors, especially those with O-negative blood, that responsibility can become visible through frequent requests to donate. Because O-negative red blood cells are especially useful in emergencies when a patient's blood type is unknown, supplies are often under pressure, a donor may be told that their blood is needed urgently, that it is rare, that hospitals rely on them. These statements are true in a practical medical sense, but repeated without context. They can sound like confirmation that the blood holds some hidden quality. Its real importance is not mysterious. It is compatible in situations where incompatible blood can kill. The most precious feature of a unit of O-negative blood is that it may enter the circulation of a bleeding stranger without presenting the most immediately dangerous ABO or D markers. The same misunderstanding can surround pregnancy prevention. To someone encountering the subject for the first time, the idea of injecting an Rh-negative mother with antibodies after delivery can feel alarming. It may be described online as interference with pregnancy, suppression of a natural biological process, or evidence that medicine is concealing a fundamental incompatibility. But the intervention does not alter a mother's ancestry or change her child's blood type. It prevents her immune system from becoming sensitized against D-positive red cells that may have entered her circulation, thereby reducing the risk to later Rh-positive pregnancies. Its purpose is not to disguise a hidden truth about humanity. Its purpose is to prevent infants from suffering a disease that once killed or injured many of them. The difficulty is that a successful prevention program erases its own evidence. When anti-D prophylaxis works, there is no sick infant, no visible crisis, no family tragedy demanding explanation. Later generations may see only an injection and a warning, not the lost children that made the warning necessary. This pattern appears throughout medicine, vaccination, sanitation, safe transfusion practice, newborn screening, and prenatal care can all become victims of their own success. When suffering becomes rare, the protective system begins to seem excessive, perhaps suspicious, because people no longer encounter the disaster it prevents. Memory fades faster than risk. In the absence of that memory, online communities can supply alternative histories. A woman asks why she received an anti-D injection and is told not merely about antibodies, but about suppressed bloodline knowledge. A donor wonders why blood services contact them so often and is told that elites require their rare blood. A person with an Rh-negative result wonders about ancestry and encounters claims about ancient gods or government databases. Each story turns a practical medical interaction into evidence of surveillance or concealment. The system's genuine opacity becomes fertile soil, especially where rushed consultations, poor health education, or past institutional betrayals have already weakened confidence. There is also a business in mystery, fear, identity, and secret knowledge attract audiences. Books, videos, private groups, personality profiles, ancestry claims, and supposed revelations can all be built around a rare trait. The people spreading such ideas are not always cynically exploiting others. Some sincerely believe what they repeat, but sincerity does not make a claim reliable. A dramatic theory travels farther than a cautious explanation because it promises transformation. It tells the reader not merely what their blood is, but why their life feels significant. A laboratory result becomes membership in a story, and stories are difficult to surrender once they have made someone feel seen. The appropriate response is not ridicule. People asking about Rh-negative mystery are often reacting to real facts. The trait is inherited, medically consequential in some circumstances, unevenly distributed, and surrounded by terminology few understand. Some also carry genuine reasons to question institutions. Treating every question as foolish only strengthens the suspicion that something is being dismissed. The better answer is more demanding and more respectful. Explain the science plainly. Acknowledge where medicine has failed. Identify what evidence exists. Admit what remains unresolved, and refuse to turn uncertainty into fantasy. What remains unresolved is worthy of attention. Scientists do not possess a complete moment-by-moment history of every Rh-negative variant across prehistoric populations. They do not know every selective pressure, if any, that may have influenced frequencies in particular times and places. Ancient DNA may yet add details to the story, but there is no credible evidence that Rh-negative blood grants hidden powers, proves non-human ancestry, or is being secretly collected for an elite bloodline. The known facts do not become less fascinating when stripped of those claims. They become more human. Behind the locked door of the blood bank, a technician selects a unit for a patient who will never see the donor. In a prenatal clinic, a mother receives preventive treatment developed from the accumulated understanding of losses once considered unavoidable. In a laboratory, a blood sample is tested not to classify anyone's worth, but to reduce danger. These are systems built by human beings, and human systems require scrutiny because they can fail. But scrutiny is strongest when it follows evidence rather than fear. The truth hidden in Rh-negative blood is not that institutions have concealed a superior lineage. It is that a rare inherited difference, once capable of bringing invisible tragedy into families, can now be recognized, managed, and used to save strangers. That may sound quieter than the conspiracy. It is also far more remarkable. because once suspicion loosens its grip, the negative sign can finally be read for what it actually preserves, not evidence of a separate humanity, but a record of the forces that shape every humanity inheritance, movement, chance, vulnerability, care, and the long effort to understand what our bodies have carried forward. After all the theories have been stripped away, after the angels, aliens, royal breeding lines, and promises of hidden ability have receded from view, the negative sign remains. It does not vanish when the mythology is removed. It becomes clearer. A person still sits in a clinic and learns that their red blood cells lack the D antigen. A donor still gives blood that may be urgently needed in an accident ward or operating theatre. A mother still receives preventive care because her immune system must not be allowed to turn a future child's inherited blood marker into a target. A population map still shows that Rh-negative blood appears more frequently in some regions and less frequently in others. The trait is not less interesting once it is returned to biology. It is more revealing, because it no longer points away from humanity toward fantasies of special origin. It points directly into the forces that have shaped humanity from the beginning mutation: inheritance, isolation, migration, mixture, chance, birth, danger, and survival. Every human body carries countless variations that began long before the life now carrying them. Some are common enough to attract no attention, others are rare, or unevenly distributed, or important only in specific circumstances. Rh-negative blood belongs to this second category. Its significance is not written across the face. It does not determine a person's intelligence, temperament, intuition, or spiritual value. Yet it offers a remarkable lesson in how human history persists below the visible surface. The absence of D on red blood cells can be passed silently through a family, preserved in a community, altered in frequency by migration, and made suddenly urgent by pregnancy or injury. It is simultaneously ancient and immediate to genetic inheritance from distant ancestors that may matter in a hospital this very morning. The first truth it reveals is that human variation does not require division into separate kinds of people. Two individuals can differ at important biological points and remain equally, entirely human. One person has D-positive red cells, another has D-negative red cells. One may safely receive certain blood that would pose a problem for the other. One may carry a pregnancy requiring additional preventive care. These differences are real, not symbolic, but they do not form a hierarchy. Human beings are filled with such differences in immune systems, metabolism, skin pigmentation, tolerance of certain foods, susceptibility to disease, the ability to thrive at high altitude, the shape of the face, the texture of hair, and the innumerable genetic details that no mirror can reveal. Difference is not an interruption of the human story, it is the substance of it. The second truth is that rarity can arise without intention. There need not be a hidden plan behind an uneven genetic pattern. Imagine again a small ancestral population, not because it possessed secret knowledge, but because most human populations were once small, a few families carry a variant. The community passes through a period of hardship, isolation, or expansion. Some family lines leave many descendants, others end. Generations later, the variant is more common than it was elsewhere. Nothing about this process feels meaningful to the people living through it. They are choosing partners, raising children, coping with sickness, moving in search of food or safety, adapting to political upheaval or environmental change. Yet their ordinary lives form a genetic pattern that can later appear strangely deliberate. Founder effects and genetic drift sound dry when reduced to terms in a textbook. In lived experience, they are the biological shadows cast by storms, coastlines, marriages, epidemics, harvests, wars, and the simple fact that some children survive to have children of their own. Rh-negative blood also reveals that populations are not fixed objects. The communities recognized today are the latest forms of histories that never stood still. The high frequencies once studied among Basque communities do not point to a people isolated from all later humanity since the Ice Age. They point to a population shaped by the same major prehistoric mixtures as its neighbours, then preserving greater continuity through later periods when other regions experienced additional gene flow. Across Europe, the wider distribution of Rh-negative status cannot be assigned confidently to a single ancient group. It may carry echoes of several events: Ice Age contraction and expansion, farming migrations, Bronze Age transformations, local isolation, and later historical movement elsewhere. Similar clinical Rh-negative results may emerge through genetic forms whose histories are not identical. What appears on the blood card as one minus sign is not necessarily one ancestral journey. This is the quiet corrective offered by genetics. Identity is not invalidated by mixture, it is formed through it. A language can remain distinctive while the people who speak it descend from multiple ancient populations. A region can preserve unusual frequencies of a trait without its inhabitants being biologically separate. A family can inherit Rh-negative blood for generations without carrying an exclusive origin. The desire for pure bloodlines usually emerges from stories people tell after the fact. The genes themselves reveal contact. Even where isolation is real, it is generally relative, shaped by fewer exchanges rather than none, by continuity rather than untouched beginnings. Human ancestry is less like a locked chamber containing one secret lineage, and more like sediment at a river mouth, layer upon layer deposited by currents arriving from different distances and different times. The third truth lies in the uncomfortable relationship between evolutionary success and human suffering. Before modern medicine, Rh incompatibility could place certain pregnancies at risk. That fact can seem puzzling. Why would a trait associated with such danger remain at meaningful frequency, especially in some populations? But evolution is not an engineer designing perfect outcomes. It works through reproduction under particular circumstances. And it is indifferent to the kind of fairness human beings wish it possessed. Where a negative status does not harm every carrier, it does not threaten every pregnancy, it does not matter unless several conditions come together: an Rh negative mother, an Rh positive fetus, exposure sufficient to stimulate an immune response, a later vulnerable pregnancy, and the absence of preventive medicine. A trait with conditional cost can persist easily, especially when carried by men as well as women. By women whose fetuses may also be Rh negative, and through populations where demographic events magnify variation regardless of whether it offers an advantage. There may yet be subtler evolutionary dimensions to the Rh system. Researchers continue to examine whether variation in Rh genes has relationships with infection, physiology, or reproductive outcomes beyond the familiar clinical account. Some suggested links remain uncertain, and claims extending far beyond evidence must be treated carefully. But there is no requirement that scientists possess a complete explanation for every shift in frequency before the larger story can be understood. Much of human genetic history is built from combinations of selection and drift, advantage and cost, expansion and loss. Some features endure because they help. Some endure because they do not hinder survival strongly enough to disappear. Some endure because the communities carrying them happen to grow. A negative blood may contain more than one of these histories at once. Its fourth revelation is medical, and perhaps the most moving. Human beings are not limited to the risks inherited from the past. For thousands of years, people carried Rh differences without knowing what they meant. Mothers lost infants without understanding the invisible mechanism responsible. Later, patients receiving transfusions could suffer reactions because medicine had not yet identified all the important blood group distinctions. None of those people lacked courage or care. They lacked information. Once scientists recognized the D antigen, the antibodies formed against it, and the danger of maternal sensitization, the ancient inheritance changed meaning. It remained present in the blood, but it no longer had to bring the same outcome. Knowledge did not erase biology. It built protection around it. There is a profound difference between a secret and a discovery. A secret is guarded by someone who wishes others not to know. A discovery is something the world has carried all along until the tools and understanding exist to see it. For a negative blood was not hidden by an ancient order, it was hidden by scale. The antigen is microscopic. Antibodies cannot be perceived by ordinary senses. Genes were carried long before humans understood that inheritance had a molecular foundation. A woman living 2,000 years ago could not have known why one infant survived, while another became jaundiced and weak. A healer could not have looked into a bowl of blood and seen sensitization. The mystery was real, but it was not a conspiracy. It was the distance between human suffering and human knowledge. That distance has narrowed. A modern blood service can identify donors, screen recipients, preserve rare units, and move blood across distances ancestors could never have imagined. A modern obstetric clinic can test an Rh negative mother, monitor antibodies, provide anti-D immunoglobulin, and follow a pregnancy with tools that transform risk into preparation. Even when complications occur, specialists can intervene in ways that would once have seemed impossible, including monitoring fetal anemia and, in severe cases, giving blood before birth. None of this makes the trait unimportant. It reveals why understanding it matters far more than mythologizing it. There is also a moral lesson in the way Rh negative blood links strangers. Rare blood does not establish superiority. It establishes dependence. A person with a valuable donor type may one day save someone they will never meet. The recipient may come from another family, another language, another background, perhaps a population in which that compatible blood is especially difficult to find. A trait made remarkable by inheritance becomes useful through generosity. Blood that online stories might use to separate people becomes, in the hospital, one of the most direct forms of connection imaginable. A red cell leaves one body, crosses an institutional chain of testing and care, and enters another body to carry oxygen through a night of danger. It does not ask about ancestry. It performs its function if it is compatible. The compatibility contains an irony. Blood types prove that biological differences matter. Transfusion proves that those differences demand cooperation rather than hierarchy. Medicine must respect the boundary between incompatible cells precisely so that human beings can keep one another alive. The lesson is not that everyone is biologically identical. The lesson is that variation makes mutual responsibility more necessary. An Rh negative patient may depend on the forethought of Rh negative donors. An Rh positive infant may depend on preventive treatment given to an Rh negative mother. A hospital depends on communities willing to provide blood, and communities depend on institutions worthy of trust. The biological fact does not isolate anyone. It binds lives together through careful knowledge. For the individual who carries Rh negative blood, the truth may at first sound modest beside the stories of lost gods or hidden powers. There may be no special gift to claim, no evidence of a secret caste, no invitation to stand outside ordinary humanity. But ordinary humanity, seen at its full depth, is not an anticlimax. In that minor sign is the survival of ancestors who endured cold, hunger, childbirth, infection, migration, and upheaval. There are populations dividing and meeting again. languages persisting or disappearing, valleys preserving local patterns, bones surrendering fragments of DNA after thousands of years. There are mothers once left without an explanation, and children now protected because their losses were finally understood. There are donors giving a part of themselves so that a stranger can remain alive long enough to see another dawn. The blood does carry history. It simply carries a history more intricate and more honest than the myth. It records no hidden superiority, but it does bear traces of who survived and how populations changed. It announces no forbidden origin, but it testifies to the extraordinary variety generated within one human species. It reveals no power of intuition or cosmic descent, but it can become powerful in the hands of doctors, nurses, scientists, donors, and patients who understand what compatibility means. The negative sign is not an emblem of separation. It is one of the countless quiet marks by which the living remain connected to the dead, to one another, and to the unending consequences of inheritance. Yet no genetic explanation can be complete if it forgets the people for whom this is not an abstract history. Behind every frequency map are actual lives, the expectant mother studying her test result, the injured patient whose type must be matched quickly, the rare donor asked to return, the family told that compatible blood has been located, the child whose safe birth rests on a simple injection and a century of accumulated knowledge. To understand Rh negative blood fully, the story must leave theories and population charts behind for a moment and return to those lives, where the enigma is not about ancient status, but about what happens when the right blood, the right care, and the right understanding arrive in time. The result arrives as a small item in a long sequence of prenatal tests. A woman who has only recently begun to imagine the future in weeks and heartbeats sits in a clinic while blood pressure is measured. Dates are checked, questions are asked about health, previous pregnancies, medications, bleeding, family history. Somewhere among the results is her blood type, perhaps O negative, perhaps A negative, perhaps another combination with the same small sign attached, before pregnancy. The information may have been no more than a line recalled vaguely from a donor card or an old record. Now it is spoken with a different weight. She is Rh negative. The words can cause worry before they create understanding, especially if she has already encountered the louder stories surrounding rare blood. But in the hands of careful medicine, this discovery is not a sentence and not a mystery meant to frighten her. It is an early warning system. One of the quiet advantages of being able to recognize a risk before it becomes harm. The appointment may seem almost ordinary. Her blood is screened for antibodies to determine whether sensitization has already occurred. If no harmful anti-D antibodies are present, then preventive treatment can be planned according to clinical practice, where she lives anti-D immunoglobulin during pregnancy. Additional doses after events that might allow fetal blood into her circulation, and another dose after delivery if the baby is found to be Rh positive. There is no spectacle in this sequence, no dramatic intervention performed beneath bright surgical lamps. Often, it is an injection, a form, a record entered correctly, a result checked at the appropriate time. Yet the normality of the procedure is the outcome of a victory over an ancient hazard. What once waited unseen inside later pregnancies can now often be prevented before the immune system begins to remember. For a mother carrying her first child, the explanations may remain strangely abstract. A clinician speaks of fetal cells, antibodies, sensitization, and protection for future pregnancies. The fetus is moving, growing, acquiring the face and hands that will soon be known intimately, and it can seem unsettling that the child's blood might contain a marker her own blood lacks. Some mothers hear the language of incompatibility and wonder whether it means their bodies are poorly made for pregnancy. It does not. Human reproduction has always involved the meeting of differences. The fetus is not a rejected stranger. The mother is not defective. Rh status is not evidence of a broken biological bond. It is one specific circumstance in which an immune system may need help not to respond to a harmless inherited trait in a way that becomes dangerous later. Many women will never experience the feared complication. They receive appropriate care, give birth to a healthy infant, receive treatment if needed, and continue with the chaotic, exhausting tenderness of life with a newborn. The blood type notation fades behind feeding schedules, sleeplessness, laundry, and the astonishing fact of a child breathing nearby. Perhaps years later there is another pregnancy, and again the records are checked. The blood is screened, the routine protection offered. The absence of tragedy may make the process seem minor, but behind that uneventful outcome stands a long history of pregnancies in which no one knew what was happening until a baby became critically ill or failed to survive. Routine medicine is often memory made practical. Not every family encounters the story so gently. A woman may arrive for care already sensitized, perhaps because of an earlier pregnancy, a miscarriage, an unrecognized bleeding event, or transfusion in a place or period when preventive measures were not available. Her antibody screening does not come back clear. The danger is no longer only theoretical. If her fetus is Rh positive, maternal antibodies may cross the placenta and destroy fetal red cells. At that point, the pregnancy may be followed much more closely. Specialists monitor for signs that the fetus is becoming anemic. Ultrasound measurements can reveal changes in blood flow, suggesting that the fetal heart and circulation are compensating for too few functioning red cells. In severe cases, the child may require a transfusion while still within the uterus. Compatible blood delivered through a procedure whose technical calm contains an extraordinary fact. A baby not yet born can receive blood in an effort to survive an immune conflict inherited through the most intimate connection in human life. The procedure belongs to modern medicine, but the emotion surrounding it is as old as parenthood. A mother may lie still while clinicians focus on images, needles, blood units, heart rate, and timing. The fetus that once seemed protected entirely within her is now understood to be vulnerable partly because of antibodies made by her own body. Guilt can arise easily, even when it has no rightful place. Her immune system has not chosen cruelty. It has responded to a molecular signal in the ordinary way immune systems respond to foreign antigens. Care is required because biology is not moral. The mother is not the cause in any human sense. She is the person through whom the child is being carried towards safety. Nearby, perhaps in a city the family has never visited, a donor has already contributed to that safety. The donor may not know whether a unit of blood is used for a fetus, a woman hemorrhaging after delivery, a crash victim, a cancer patient, or a patient undergoing major surgery. The act of donation is usually separated from its result by privacy and distance. A person sits in a reclining chair, answers screening questions, feels the brief discomfort of a needle, watches a collection bag fill, then leaves with a bandage on the arm and the rest of the day ahead. Yet for someone with O-negative blood, or with another particularly useful compatibility profile, the donation may be sought repeatedly. Blood services know that the demand is constant and that supply can thin quickly. A holiday period, a storm, an epidemic, or simple lack of donors can leave hospitals balancing scarcity against emergencies that refuse to wait. The O-negative donor is sometimes described as a universal donor, a phrase useful but requiring care. O-negative red blood cells are often used in urgent situations because they lack A, B, and D antigens, reducing the most immediate risks when the recipient's type is not yet known. But transfusion medicine is more complex than that phrase suggests. Other blood group antigens matter. Plasma has different compatibility considerations. And whenever possible, clinicians move toward type-specific and fully matched blood, rather than drawing indefinitely on limited O-negative supply. Still, when an unconscious patient is brought through the emergency doors bleeding heavily, O-negative units can mean survival before identification and full testing are complete. The rare donor's blood does not carry magic, it carries minutes. Those minutes are never theoretical to the trauma team. A motorcycle collision, an industrial accident, a ruptured vessel, a violent injury, a sudden obstetric hemorrhage blood loss can turn the strongest body frighteningly fragile. Skin pales, pulse rises, consciousness slips. The heart tries to maintain pressure as circulation empties. In massive bleeding, medicine must act while questions are still unanswered. Who is this patient? What is the exact type? Are there antibodies from earlier transfusions or pregnancies? What injuries remain hidden? Blood is released because death will not pause while every detail is settled. A bag labelled O-negative may be lifted from storage and carried toward a person whose name the donor will never know. For an Rh-negative patient, finding safe blood becomes even more important after the initial crisis, particularly if the patient is likely to form anti-D or already carries antibodies. A woman of childbearing potential may be protected from unnecessary exposure to D-positive blood, because that exposure could complicate future pregnancies. A patient transfused many times for a chronic illness may develop a complex antibody profile, turning each later transfusion into a more demanding search for compatible units. The blood bank staff who conduct that search work behind the visible drama of the hospital, but their decisions are as crucial as any instrument in the operating room. They inspect histories, repeat tests, identify antibodies, call other centres, locate reserved or frozen units, and calculate how to support a living body without triggering another danger. Sometimes the search crosses borders. Rare blood donor networks exist because certain compatibility needs cannot be met by the nearest hospital or even within one nation's routine inventory. A patient with an unusual combination of missing antigens and existing antibodies may need blood from a donor whose cells match in exactly the right way. Units may be preserved frozen for years, released under strict conditions, transported urgently, and transfused into a person who had begun to run out of ordinary options. O-negative blood is only one part of this wider world of rare compatibility, but it introduces many people to the principle what is uncommon in one person can become invaluable to another when medical need is precise enough. There is a humility in the rare donor registry that the mythology misses. A donor is not entered into it because the blood marks them as more evolved, more ancient, or more powerful. The registry exists because human bodies differ, and because those differences can make care difficult in an emergency. It is an archive of possible help, a name, a type, a tested profile, a contact route, a willingness to answer when the request arrives. The donor may be a teacher, a mechanic, a student, a parent. A retiree, someone whose ancestors and beliefs have nothing in common with the recipient. The compatible blood crosses every narrative humans invent to divide themselves. For the hours it matters, the most important shared fact is not prestige or lineage, but that one body can sustain another. The mother in the prenatal clinic and the trauma patient in the operating room appear to occupy different stories. One is preparing for birth, the other fighting against sudden death. Yet Rh-negative blood links them through the same principle of foresight. A test performed before crisis allows harm to be prevented. A donor recruited before shortage allows blood to be available. A medical record preserved accurately allows a future team to avoid an antibody reaction. A registry built patiently allows a rare match to be found when someone is already in danger. None of this offers the instant drama of secret histories, because its deepest achievements often look uneventful, the baby who was born healthy, the patient who survived surgery, the transfusion reaction that never occurs, the unit on a shelf waiting exactly where it is needed. In regions where preventive care and blood supplies are limited, the old dangers become harder to forget. A mother may not learn her Rh status early in pregnancy. Anti-D immunoglobulin may be unavailable, unaffordable, or inconsistently supplied. A hospital may face shortages of compatible blood, particularly in places where Rh-negative donors are uncommon, or where donor systems struggle under wider resource pressures. A child may suffer a preventable disease not because science lacks an answer, but because the answer has not reached every family equally. The global story of Rh incompatibility is therefore also a story about access. Knowledge can exist and still fail to save unless health systems, transportation, donor participation, screening, and affordable care bring it to the person in need. No single explanation fully accounts for every loss in such settings, and not every newborn anemia or pregnancy tragedy involves Rh disease. But the injustice is plain when a well-understood, often preventable condition persists, because care is unevenly distributed. Here the language of hidden truth takes on a different meaning. The truth is not being concealed in a secret archive. It is known, published, taught, and routinely used in many hospitals. What remains hidden from some lives is the benefit of that knowledge, the test not performed, the injection not available, the specialist too far away, the compatible blood unit that never arrives. The gap is not between ordinary humanity and a superior bloodline. It is between what medicine can do and who is able to receive it. A healthy newborn held against a mother's chest offers no visible sign that this history has passed close by. The child's tiny circulation carries inherited antigens. The mother's blood carries its own genetic absence. Around them are the results of generations of inquiry testing, preventive antibodies, donor systems, clinical vigilance. Outside the hospital, a donor who gave blood weeks earlier may never know that their decision became part of someone else's survival. This is how the rare bloodline truly acts in the world, not as a coded inheritance of hidden beings, but as a practical connection among vulnerable lives. The ancient enigma has not vanished. Questions remain about the distant spread of Rh negative variants and the precise forces that shape their distribution. But for the people who face the trait in the present, its most urgent meaning is no longer buried in prehistory. It is here, in an injection given before antibodies form, in a blood bag released before circulation collapses, in a donor who answers a call, in a baby whose life begins without the shadow that once followed later birth through a family. The mystery has travelled far, through ice and migration and myth, only to arrive at something profoundly simple: understanding blood matters because people matter, and sometimes the difference between inheritance and tragedy is whether care reaches them in time. The newborn leaves the hospital without knowing that an ancient question has followed it into the world. The wristband is removed, the records are stored, the mother, tired and watchful, is given instructions, reassurance, perhaps one final confirmation that preventive treatment has been completed. Outside, traffic moves, doors open and close. Families pass one another without knowing whose blood carries D and whose does not, whose cells might one day be urgently compatible with a stranger, whose ancestors carried a particular absence through migrations no history ever recorded. The child is not the answer to the Rh-negative enigma, nor the beginning of it. It is simply the newest bearer of a human inheritance that has travelled through countless lives and continues onward, quietly, without announcing all that remains uncertain behind it. There is a temptation at the end of any mystery to demand a final unveiling. One image, one forgotten tribe, one mutation in one remote valley, one ancient population from which all Rh-negative people descend. A story that began with the absence of an antigen seems as though it ought to end with the discovery of its precise first carrier. But biological history rarely offers conclusions so clean. The Rh system is old, complex, and shaped by more than one kind of genetic variation. Across different populations, a person can lack detectable D antigen through genetic backgrounds that are not perfectly identical. Among many people of European ancestry, deletion of the Rhd gene is a central explanation for Rhd negativity. In other populations, altered or inactive versions of the gene and more complex arrangements may also produce a negative or apparently negative phenotype. The same sign on a medical record can therefore represent histories that overlap without reducing to one origin. This does not mean nothing is known, far from it. Medicine knows with great reliability why Rhd status matters in transfusion and pregnancy. Genetics knows where the principal genes involved in the Rh system are located, and how many important variants affect expression. Population studies know that Rh negative frequency varies markedly among regions and ancestral backgrounds. Ancient DNA has reconstructed enormous movements that form the setting through which blood group variants must have travelled. Research has shown that the Basque people, once treated in popular imagination as a sealed Paleolithic remnant, are better understood as a population with substantial continuity after the major prehistoric formation of Iberia. Genetic evidence has shown that modern humans carry ancestry from Neanderthals, while providing no credible support for the claim that ordinary Rh-negative blood is a unique Neanderthal marker. On the central questions of medical reality and human belonging, the picture is clear. The remaining uncertainty lies deeper in time. When did the most common Rh-negative associated forms arise? In which ancestral populations were they first frequent enough to influence later distributions? Did one major prehistoric demographic event contribute disproportionately to the high D-negative frequencies found in parts of Europe? Or did the pattern emerge through several expansions, bottlenecks, and periods of isolation? Did natural selection play some role in particular places or eras? Perhaps through effects still not fully understood, or is much of the distribution best explained by genetic drift and demographic history? Ancient genomes may eventually give firmer answers, but the record is scattered, damaged, and never fully representative of everyone who lived. There may never be a single day on which the mystery is declared solved. Science often works by narrowing possibilities, strengthening some explanations and weakening others, rather than producing a final dramatic revelation. A newly sequenced ancient genome can place a variant in a particular region at a particular time. 10 more genomes may show whether that individual was typical or unusual. Samples from a later age may reveal that frequencies changed after migration or population turnover. Genetic modelling may suggest likely roots, then require revision as new remains appear from places once poorly represented. The story grows more exact by becoming more complicated. It does not become less truthful, because it lacks the neat ending of a legend. What can be said with confidence is that the absence of D belongs fully inside human evolution. There is no evidence that it was inserted into humanity by visitors from another world. There is no evidence that it identifies the descendants of angels, gods, or a hidden ruling caste. There is no evidence that it provides psychic gifts, superior intelligence, special perception, or an inherited claim to greater importance. The unsupported theories fail not because they are too imaginative for a dull subject, but because the real evidence points elsewhere toward ordinary genetic variation, passing through populations whose histories are far more interwoven than myths of special blood permit. The truth is not that Ro-negative people stand outside the majority of humanity. The truth is that any attempt to define ordinary humanity too narrowly will fail. Human bodies contain variations shaped by deep ancestry, environment, random inheritance, ancient meetings, ancient separations, and the uneven expansion of family lines. Some traits are common in one region and unusual in another. Some become medically significant only when two bodies meet in pregnancy or transfusion. Some remain mysterious at the level of evolutionary origin while being thoroughly understood at the level required to prevent harm. Our negative blood is not an exception to what it means to be human. It is one of the clearest examples of it. This may be why the subject continues to attract people even after the most sensational answers are set aside. Beneath the false stories is an intuition that is not false. Blood does connect the living with a past they cannot remember. It does carry evidence of journeys older than nations, older than languages still spoken, older than written names. A person looking at a blood type result is not wrong to feel that inheritance has brought something ancient into the present. The mistake is assuming that ancient must mean exclusive, or that rare must mean elevated. Much of what is ancient inside a person is shared in unpredictable arrangements with strangers. Much of what has survived did so not because it marked greatness, but because human life continued. Somewhere in the ancestry of every Rh-negative person are individuals who carried the trait or the genetic potential for it without ever knowing. Their worlds were different from one another. One may have stood on a coastline now beneath the sea. Another may have crossed pasture with animals during the first spread of farming. Another may have lived among Bronze Age settlements while languages and family structures changed. Another may have prayed in a medieval village for the survival of a jaundiced infant, unaware that antibodies were passing through the placenta. Another may have arrived in an industrial city, fought in a war, crossed an ocean, raised children through scarcity, or walked into one of the first hospitals where blood typing could give a name to the inheritance. The chain is not noble or ignoble. It is human continuity, with all its losses concealed within successful survival. The same is true of Rh-positive people, and of every blood group. No person stands outside the flow of ancient accident and inheritance. The plus sign is not normality in any moral sense, just as the minus sign is not mystery in a supernatural one. The categories are clinically meaningful because medicine must pay attention to differences. But outside the moments when compatibility matters, the body offers no reason to rank one over another. In a hospital, a rare donor can save a common recipient, and a common donor may save a rare one, depending on the exact need. Blood systems make interdependence visible. They reveal that biological difference is not a verdict on belonging. It is a reason for knowledge, preparation, and care. There is another hidden truth in the story, one less likely to be advertised in dramatic titles because it offers responsibility instead of secret status. A person who knows they are Rh-negative can act on that knowledge. They can understand its relevance during pregnancy. They can ensure clinicians have accurate records before planned surgery. If eligible and willing, they can donate blood, perhaps repeatedly, because their compatibility may matter urgently to someone else. They can resist frightening misinformation that turns preventive care into suspicion. And they can speak about rarity without turning rarity into a mythology of superior or separate people. Information inherited passively can become useful through deliberate action. For medicine, the lesson is equally direct. Facts are not enough if they are delivered without clarity or respect. A patient learning that their blood type affects pregnancy deserves an explanation that does not leave them feeling defective or alien. A donor repeatedly called because their blood is valuable deserves to understand why. Communities whose trust has been damaged by medical misconduct deserve transparency, ethical accountability, and access to care, not dismissal. A subject surrounded by conspiracy cannot be answered only by saying that conspiracy is false. It must also be answered by making the real science comprehensible and the real institutions worthy of confidence. This is particularly urgent because the remaining suffering is not mysterious. In places where screening, anti-D immunoglobulin, trained care, and compatible blood are reliably available, Rh-related disease has been greatly reduced. In places where those resources are missing or uneven, mothers and infants may still face preventable danger. The most important unresolved struggle is therefore not discovering whether a secret bloodline rules from the shadows. It is ensuring that known protection is available wherever it is needed. A child should not face an old immune hazard simply because geography, poverty, conflict, or failing health systems kept prevention out of reach. The story of Rh-negative blood is not complete while scientific knowledge exists in one place, and avoidable loss continues in another. Back in the blood bank, shelves hold units marked by type, collection date, testing, and careful handling. Nothing about them resembles a relic from a vanished world. They are ordinary bags of dark red fluid, quiet under refrigeration. Yet each one carries a history beyond documentation, the donor's inheritance, the donor's decision, the medical knowledge that identifies compatibility, the patient who may soon require it. On another floor, an infant sleeps near a mother whose chart carries the same negative sign that once invited fear. The treatment has been given. The risk has been understood. A danger that shadowed human reproduction for millennia has been made smaller by knowledge and cooperation. The enigma remains, but it has changed shape. At first it seemed to ask what strange origin might be concealed within rare blood. Now it asks something more difficult and more honest. How many lives are carried inside one inherited variation? How much history can pass invisibly from parent to child? And how often humanity invents fantasies of separation when the evidence reveals dependence instead? Whereas negative blood holds no proof that some people are other than human. It holds evidence that being human has always meant carrying difference through danger, crossing paths with others, and surviving not in isolation, but through the fragile continuity of bodies that protect, give, receive, and begin again. The child grows, and the blood type recorded at birth becomes one detail among thousands that shape a life without defining it. There will be first steps, scraped knees, illnesses passed through, seasons remembered only by photographs, perhaps journeys far from the place of birth. The quiet inheritance remains within the circulation, carried from heartbeat to heartbeat, asking for nothing unless one day a pregnancy, a donation, an accident, or an operation brings its meaning briefly into view. Then the old thread rises again, not as a sign of destiny, but as a reminder that every body is both private and connected. Long before laboratories could name the absence of a single antigen, human beings watched blood with awe and fear because they knew instinctively that it marked the boundary between life and loss. They could not see the cells, the antibodies, the genes, or the ancient movements preserved in their distribution. They knew only that blood was warm, that it passed from mothers into children in ways they could sense but not explain, and that when too much of it left the body, the person followed. Across the millennia, knowledge has drawn closer to that mystery. It has not made blood less meaningful. It has replaced terror with understanding wherever it can. There are still missing pages. Somewhere in the unreachable past, genetic variations arose in people whose names no one will ever recover. They crossed rivers, coastlines, grasslands, and mountain passes. They sheltered from winter winds. They buried infants. They joined strangers. They survived long enough for something silent in their blood to continue. Their descendants became other peoples, spoke other languages, forgot the roads that had brought them there. Eventually, the invisible difference reached the modern world, where a drop of blood beneath laboratory light revealed what centuries of grief and survival had never been able to say aloud. The secret was never that certain people were less human, or more than human. It was that human beings have always carried more history inside them than they can see. A minor sign on a record can contain the shadow of migrations, the memory of vanished communities, the vulnerability of birth, and the kindness of a stranger who gives blood without knowing whose life it will enter. And so the story ends not with a throne, a spacecraft, or a sealed archive, but with something smaller, a mother holding a sleeping child, a donor's bandaged arm, a refrigerated unit waiting in silence, red cells moving through darkness as they have always moved, bearing oxygen, inheritance, and the fragile unbroken passage of life from one human body to another.